help if I unmuted the intro song. All right, everybody. Thanks for the Growing with Fishes podcast, episode 265. Hard to believe we've done this many. We are back with one of our favorite guests. It's been, over 100 episodes, I think, since you've been on the show. Thank you so much for joining us this evening, Dr. Efron.
Hello, Steve. Good to see you again.
Always glad to have you on the show. You're certainly one of the most knowledgeable people that especially on the topics of mycorrhizae and just general soil science. So super excited to have you back on the show. We also have Fumidor with us as well. Thanks a lot for joining us.
How's it going? Cheers. Good to be here.
Hey, Fumidor.
Nice to meet you, Efren.
Yeah, we follow you on
Cheers.
Instagram. I see that.
Right on. Cheers.
Fumidor.
Awesome. Well, we have a great show for you guys tonight. If you guys haven't already, be sure to go over to apmjclass.com and check out the class that Marty and I put together. If you guys haven't already, you can use coupon code APMJ now through Cyber Monday to save $50 off the class if you're interested in that. Alrighty, and if you other thing, housekeeping notes real quick. If you guys haven't already, please go back and check out the second annual virtual Aquaponic Cannabis Conference. I'm going to release first the live on YouTube. Hold on. Yeah, no, we're live. It's good. Sorry. I'm going to release the stuff that got cut off at the end of the 12-hour block on YouTube first, and then we'll release the rest of the content after that as I edit it up. So if you guys didn't already check out the conference, we have 28 hours of conference content from last weekend with over 30 different speakers from around the world on aquaponic cannabis, the science behind it. There's wonderful, great breakdowns by a couple of different universities and companies on side-by-side comparisons between living soil, compost, and aquaponics, and every combination thereof. It really is a great conference and tons of awesome speakers. So definitely go back and check that out from last weekend. All right, thank you so much for joining us, Dr. Efron. Maybe give us a little bit of a quick intro for those of you that missed the first episode and don't know much about you, and then we'll get into some of the questions. You are quite the expert. For those of you that don't know, Dr. Efron is probably the world's leading expert in mycorrhizal fungi, especially when it comes to our favorite plant. So I'm super excited to have him on the show tonight.
Just a little bit. I can't remember what I said 100 episodes ago.
Oh no, it's perfectly fine. Maybe talk with a little bit about your background and it could be a long story, but I trained as a biologist, in the early 80s or mid 80s. Then I got my PhD in botany and plant pathology here at Oregon State University. Almost 30 years ago, 92. So just getting there. So I've been, I do a lot of fungal taxonomy, fungal identification for the Forest Service, especially rare and endangered species, mainly corals and truffles and some other groups, coral mushrooms. I do a lot of mycorrhizal assessments, pretty much of any plant. I can recognize all different types of mycorrhizae, mainly ectos and arbuscular and ericoid. And those are the main ones that most people see in agricultural crops. I do also spore extraction or arbuscular mycorrhizal fungi from soils, which is very important to know what the spore numbers in your soils. The soil bank, how much, how it's kind of we use it as an indication of how healthy your soil is. If mycorrhizal fungi are actively, producing and leaving spores in the soil, it's an indication, that things are going well. If we see that the spores numbers are pretty low, something must be happening, it's sort of like a little bit of a clue. And so when we put those numbers together with colonization percentages throughout your area or your acreage or your system, and then
we get another clue. If we have high levels of colonization in a great spore bank, that means something is really happening in the soils. Something positive is happening in the soils. So that's kind of how we do that. And we're working a few projects. They're starting the Obama Presidential Center in Chicago. We're salvaging soil over there and trying to recover, the beneficial mycorrhizal fungi so we can use it in the new landscape. We're actively doing that. We're going to start a new project in LA, a wildlife bridge, the world's largest bridge, to sort of help the cougar population because they're inbreeding, because they keep getting killed trying to cross 101, the highway. And so the Santa Monica Mountains are split by the 101. And so the private funding got the money after, I don't know, maybe 10 years of searching. They got the funding to get the project going. And so we'll be doing some soil biology over there and using mycorrhizal fungi to restore the area and so on. Practical application type inoculation, create nurseries, and native plant communities and such. And so those are the two. Some other in Houston. We finished Expedia campus in Seattle. Doing some others in Houston. We're still helping the Cowboys, Dallas Cowboys, on the practice field to keep the evaluation of the practice field because they inoculate mycorrhizal fungi. So it just keeps me entertained, clients here and there. And
So I'm an Eagles fan. Can I give you some recommendations on what to do to their field?
Yeah, well, if they use natural turf, yeah, we can do something with them.
Oh no, I meant more about screwing up Dallas.
I see.
I'm super happy to have you back. I think the first question I would love to have you talk about, maybe before we even get to that, is do you want to tell us maybe a little bit about MycoWeb and some of your current work and that people might not be aware of, or some of your current services that people are aware of or unaware of that you provide for the community? Because I know you work really heavily with a lot of cannabis growers.
Well, the services that I provide is, the practical stuff. I do mycorrhizal assessments of roots of any crop, and for the cannabis, and especially if you grow them in soil, obviously, you're gonna, and if you don't inoculate with commercial mycorrhizal fungi, you're gonna pick up, whatever is in the soil, and so, assessing the roots that will give you an indication of, how active was the mycorrhizal fungi in your soils, and as well as spore banks. If you don't inoculate and you grow it in pots, well, it's kind of a wasting of money because it's unlikely you're going to have mycorrhizal fungi unless there's some natural soil there. So that's kind of my take. So don't waste your money. If you inoculate, there's no point unless you grow them in pure soil. The spore counts, again, if you grow cannabis directly in soil, well, we can test your soils, and I only need a cup of soil. But again, I can't stress this enough. A lot of people, well, they may not have a clear understanding how mycorrhizal fungi works. They're not even in the soil, they're patchy. And so it's just the nature of the fungi, the life cycles. When they're patchy in their soils, one sample is not going to tell you the whole story. You're just testing that spot. If you move a foot away or 3 feet away, those numbers may change. So if you have acres and acres of soil, sampling one piece of soil, you just gotta take the data with, with grain of salt. Don't make big management decisions based on one sample, because you can have pretty low numbers of spores on that sample for whatever reason, but the rest of the soil may be good. And so if you make decision based on that sample that was so low, maybe you're wasting your money, or it's just the wrong decision. It could be vice versa. If you got one sample there's pretty high numbers for whatever reason, that doesn't mean the rest of the soil is also high, unless you have a history of growing the same crop, and then you have, so you gotta think a little bit of what are you going to do with the data you get out of your test. Replication is really, really important, and that's what statisticians they keep telling everybody. If you want to make decisions based on statistical analysis, you got to have enough sampling of the population or whatever you're measuring, because otherwise, it's just not going to be significant. And if you're not going to compare, detect variation within your system, again, you're not going to make good decisions, or you're just going blindfold. And I understand, doing all this analysis can be very expensive, but at the same time, you don't want to waste money when maybe your cells are just good, so it all depends on how solid you want to do your management recommendations, on a good guess, or based on data.
And having the right the fungi can really make a huge difference in yields and plant health, especially. I noticed, especially going from dual root zone with mycorrhizal fungi to straight aquaponics, it's not even a comparison. It's not even in the same league in terms of how much better the plant's immunity is with that soil level present and having some level of mycorrhizae impacting the plant's immune system, which I know that we'll get into a little later on, but certainly something that I've observed firsthand, and I think that is super important. One thing that I think a lot of people often are told over and over again that I learned was maybe not as true as I thought it was, from you specifically, was that a Rhizophagus irregularis really, traditionally is told to you as the only mycorrhizal fungi that can colonize cannabis, and you were, I think, the person that discovered that that's not true. And I'd love for you to talk about that.
Yeah, I mean, unfortunately, there's let's see a little bit of background. There's probably about, I would think, about 300 species of arbuscular mycorrhizal fungi only known, but there's could be thousands of them that we just don't know what their names are, especially from the jungles or the desert extreme environments, because the species that we see in commercial products, those are the most common species, and those were isolated from different areas. I mean, the original species were isolated from different areas. For example, the famous Rhizophagus interradices, the most common commercial species. That species was originally isolated from orange trees on an orchard somewhere in Florida, and that species, the original species, they will keep propagating it, multiplying it, and that species gets spread, to another grower or to a culture collection, and so that species is all over the place. Now, there could be other intraradices isolates from different areas, from California, or isolated from a banana plantation, or from some potato field in Idaho. So that species is cosmopolitan as far as we know. And so that species as a name has been used a lot in commercial products. Now, one strain, or one strain or isolate of that species could behave completely different than the same species but isolated from a banana field. And so even though they're the same species, they may not function the same way. They may not be adapted, especially, after so many cycles run, by that culture. And so that's just one species. The same goes for Glomus mosi. It's another one very common. It's called now Funeliformis mosi. And then we got Rhizophagus irregularis, which is another species from that is also isolated from Canada, from Chile, from Europe, from Mexico. It's all over the place. And so we don't know how many species are actually the ones that create a positive effect on cannabis. Now, the question is, or the hypothesis is, as one species of Robuscula mycorrhizae effective with all varieties of cannabis that we know now? Nobody knows, because the original cannabis species has been, modified so much that, and again, here's an hypothesis. We might be dealing with an organism with cannabis species, the new strains that they're being developed constantly, yearly. There's so many varieties. Unless you test them with the same species over and over, we're always going to be speculating that that one species fits all. And that creates an issue, because people keep buying products. Yeah, but is the product is not pure. The product has one species. The product has a variety of species, at least 4 or 5, and it has bacteria, and it has other little nutrients. So when you apply it, you just don't know what is causing the effect. What species is causing the effect? Are the 4 species actively, affecting or influencing positively the cannabis at the same rate? How about the bacteria? Are the bacteria doing the positive effect right away? Because here's another thing. Bacteria are active almost immediately as they touch the soil, because they're constantly growing. They're multiplying exponentially. One bacteria splits and then makes it 2, and another one makes it 2 and 2. And so it's exponential growth. And so if they have a great habitat in the soil, they're growing, they're already affecting the plant. And what did we say? Oh man, this product is amazing. Mycorrhizal fungi is amazing. Wait. The spores of mycorrhizal fungi, they're dormant. They take a little bit of time. It's just like when you feed a cat and a dog, the cat is just going to stretch and look at the food, and the dog is just immediately, chowing down the food. So they're working at a very, very different speeds, because they're totally different organisms. They have totally different life cycles. And especially biologically, one is a tiny 1 micron organism, compared to a fungal hyphae. You have a spore that is dormant, and that spore measures, let's say, between 500 and, between 50 and 100 microns. That spore it has all the energy inside, all the nutrients, the energy is inside. Once the spore germinates, it's using the energy from the spore to grow a hyphae. The hyphae needs to grow that filament. And then if they're lucky, and if they find a root available to colonize, and if the root is willing to, the lock and key concept, if they open the door, the hyphae attaches to the root and then penetrate the root cell, and it start growing inside the root system and the cortex cells, and it starts spreading. that takes a lot of energy. But now the switching from the energy from the spore that are left behind, that spore gets dissolved and destroyed. Now the fungal filament is growing and living out of the sugars from the roots. And they're taking their time to grow. And so the hyphae grows in the soil, what is growing in the root system at the same time. And so once the hyphae is starting to attach to the soil particles, that fungal hyphae needs to be in close contact with the substrate, because they need to absorb, on a solution, water, the nutrients through the fungal hyphae. It's just like a little filter. It goes to the cell wall. The nutrients goes to the fungal hyphae inside that cylinder, that microscopic filament. And the nutrient goes inside the cell, the fungal hyphae inside the cell, because it's the same individual, and then transfers it to the plant to feed the plant. So let's say phosphorus, orthophosphate, the most simple thing, because the phosphorus needs to be at the smallest size possible molecularly, because it has to go in solution through the cell wall of the fungal hyphae. Otherwise, it's not going to go through that wall. So the simpler the phosphorus, the better chances to go through it, absorbs it and transfers it. And then the sugars they're finding on the plant are the ones that are being used by the fungus to grow and create more cell biomass, and it just keeps growing. Now we're talking a period of 2 or 3 months of growth. And the bacteria by that time completed many cycles by then, and so they're working at a total different speed. So the product might be working, but may not be the mycorrhizal fungi right away. So don't jump quickly, usually with the same amount of cannabis, to see what species of bacteria are affecting better, the cannabis, one at a time. Now I'm going to test the fungal species, one at a time. Then you're going to be able to pick up which one is the one that causes the most effect. Now, that's on that side. The problem or the challenge is, what are you going to measure to show efficacy? Are you going to measure biomass? Are you going to measure nutrient content in the tissues? Are you going to measure CBD production, or oils on the flower, or flower numbers, size of leaves, diameter of the stem? What are the parameters that are going to indicate efficacy by that species? Biomass, root volume, there's so many parameters that you can measure, and not all the parameters will be easy to measure. And so it creates a big experimental design. And once you get a big experimental design, that means you have a lot of replication, and the whole thing becomes a monster, and it's very expensive. And there's no funding for that, because the growers, they just want to grow the cannabis, their strains, and done with it. So there's no time for science. So we only do a little bits of testing, kind of like a let's just eyeball it. Yeah, my plants look a lot better, but that's just an observation. That doesn't work in science. It's just an observational. But at the same time, the actual grower is the one that knows the most about their plants. And so their opinion is highly valuable, because they're the ones that do all the applications and all the inputs, and they're the ones that could develop so many hypotheses for the scientists. If they have a clear understanding how to test on a scientific way, on a statistical way, which is not very cheap. And so either you grow cannabis to produce your flowers and done with it, or you want to do science? Because if the cycle on that year doesn't go as you expected, well, you lost 1 year. You know, the experiment, it has to be as well designed as possible, so the data is solid at the end of the experiment. And so the design has to be done very carefully to answer the original hypotheses. And the hypothesis has to be well stated. The design has to follow the hypotheses to make sure that the answer is going to be, the question is going to be answered with a yes or no, because that's the hypothesis. You know, does this species cause a positive effect on biomass? Yes. The hypothesis, the alternate hypothesis, does this species cause a positive effect on biomass? No. And so what is that going to, you know, that's how you're going to answer the hypothesis statistically. You know, how many plants show positive effect? How many plants did not show an effect? Any variation? Was it like, you know, night and day, or they're pretty close? And, you know, and using the controls, you know, the good controls for each hypothesis, because without the controls, you know, there's nothing. So, but again, so we got Rhizophagus irregularis, Rhizophagus interradices. Those are the new names. Funeliformis mossy, Septoglomus deserticula, and Rhizophagus itunicatum.
Efren, how's it going, buddy?
Hey, Josh.
I wanna ask a question. So I heard there's this study that came out in 2018 on probiotics, right, for the human biome, right? You know, and it was, in the study was, so a person takes antibiotics, and one person does nothing, and they just let their body recover naturally, and the other person takes probiotics, which we all know is mostly Bacillus species. And the study said that the person that did nothing ended up having a better biome, an overall diversity, where the person who took probiotics and was blasting their system with the bacillus, they ended up having a limited biome. So I was just kind of saying that to say, like, when people ask me about, you know, using Mycorrhizal products, you know, some people say, well, it's in the natural environment. Why do I need to buy a product? And I guess I was just kind of throwing that out to hear your response.
Yeah, no, it's a valid question. Again, we go back exactly what we're talking with Steve at the beginning. If they say, well, it is in a natural environment, the first question is, did you look at it? You're assuming that it's there. So that's the first assumption, that we're just going blind. It's like, okay, I'm assuming that it's there. Well, we don't know how much is there. And then are the species in the native soils compatible to cannabis? That's the $64 million question. What are those species in my native soil will be compatible with the cannabis? Now, it's not going to be easy to find that out, because you can put them on your soil, right? You cannabis. You don't apply anything. Yeah, you check for mycorrhizae at the end of your cycle. Yes, they were positive. Okay, we have mycorrhizae. We don't know. We can't identify mycorrhizal species based on colonization. There's just no way. The colonization can be done by many species at the same time. So whatever is in your soils actually went into cannabis individuals. Another question is, how many of your, let's say 100 individuals got colonized? Did all your 100 individuals got colonized? Well, that's going to be a hard test, that, because that's a lot of money and a lot of time. But we can do some good sampling, random or targeted from different areas, and see how many of those individuals got colonized. And so now we know that whatever is in my native soil can grow with cannabis, but we don't know which species. And so the key is to isolate the species that are in your native soils and find out after I don't know how many cycles you can do that to find the species that will grow in your soils. That's kind of the overall thing on how to apply mycorrhizal on your soils. You gotta distinguish between speculation and for certainty. Yes, because I test my soils and we got fungal spores, lots of them. Okay, and let's just hope for the best. Grow your cannabis and check your cannabis to see if you actually connect them. If they did, now you got a bingo. Who cares which species? It works. It works on your soils. Now, if you add mycorrhizal product to your already native pool, it is again, it's an hypothesis. It is unlikely there's going to be any competition. There's just going to be like N+1. So you're just going to add more to the spore bank, because the more spores you have in your soil, the higher the probabilities of colonization you'll have. Because not all spores survive. Not all spores will germinate. And the ones that germinate, not all they're going to colonize the root systems. So, the numbers are starting to get smaller and smaller as the cycle keeps moving. And so obviously the more the more tickets you buy for a raffle, the higher the chances to win, right? It's all probabilities. So,
So I mean, just to follow that, I mean, they're reproducing on some level, right? If let's assume we have colonization on our cannabis roots, and I'm doing all the proper things, and I'm planting, another plant right next to the root. A couple of weeks after I pulled the other plant, we can almost assume that it's there. But given that, would you still see benefit to dumping down more spores to give more chances?
Yep. I would. Yep. But again, the dosage will be a lot less,
right?
If you know that you got a plenty spore bank. They say, well, instead of the recommended dosage, I'm just gonna use half of it because I got plenty. I'm just gonna use half of this. But if your soils are really beat up and your spore banks are really slow, very small, so the numbers are pretty low, you're going to benefit regardless. Increase the spore bank. And again, regenerative agriculture, without knowing, is probably what you're doing, because all those cover crops, hopefully there's no brassicaceae in those cover crops. All those cover crops, what you're doing is just activating whatever is in the soil. And by activating is waking up the mycorrhizal fungi in that soil, keep them active, keep them alive, keep them active during the slow months, the winter months. And so when you mow those things, you're leaving all the spore bank, in the soil. That's what is likely to be occurring. If you want to have data, well, you check what was the spore bank number at the beginning, before cover crop, and then what's the spore number after the cover crop. And then, but you got to make sure you list the species what you use. So let's say I'm going to use 3 or 4 different cover crop species, and then find out if the spore banks went up. And then I can use these cover crops again because they work for me for this particular objective. Hopefully it will work for you for other objectives that you may, obviously if you put a clover in there, a legume, you're bringing nitrogen to the system. And also they're mycorrhizal host. So there's a double bang in there for your dollar. You got or vetch, that's a good one too. Or flax is also a good one. A daisy flower, whatever grows, and they don't have to produce flower to benefit the thing, as long as they produce roots. And even weeds, if you think about it, even if you have weeds, some of those weeds can have some very good mycorrhizal colonization. You just got to take care of, the tops. Eventually, don't eradicate them. Be friendly, because a lot of those weeds also leaving spores behind. The mycorrhizal fungi don't care. Their weeds are the ones growing, but we do.
Yeah, the only definition of a weed is a plant that humans don't see utilization in. So if it's not medicine or food, then we call it a weed.
Dandelions are awesome. Their dandelions are awesome for mycorrhizal colonization.
It's interesting. I never heard about dandelions being good myco-friendly things, but it makes sense considering how big their root structure is.
Yeah, and pretty much any species of the daisy flower, I've seen such a good colonization. Anything on the sunflower family.
So that's a really interesting. So one of the other things that I thought was really a great takeaway from the first time I heard you talk was about, just to further support what you're talking about, is having those companion plants really has a place for those mycorrhizal fungi species, maybe even ones that won't directly colonize cannabis, to still allow it to exchange nutrients and other important compounds that the plants need to succeed. And I always love to describe it as kind of like a stock exchange, where sugar is kind of the cash, and all the other commodities are all the other things that, nutrients and secondary metabolites and amino acids and all the 10,000 things that your microbes exchange. And do you want to talk maybe a little bit about that and the importance of the different species interacting together? You kind of touched on it there, but kind of why it's so important to have those.
You can get creative, and again, the growers, like Josh and others, they're the ones that know what works, and it's very likely, you've seen plant species showing up in your soils without planting them, right? They show up and there's how do you get here? Well, they're looking for food, right? They're resources. There's mycorrhizal fungi, there's bacteria, there's good nematodes, and they're just landing on a spot, and they just happen to grow there. I've seen a lot of growers using marigolds, which is an amazing plant also to have as a companion plant, because they have apparently they produce substances in the root system that takes care of predatory nematodes. They keep down the nematode population, and also they exudate some substances. They act like insecticides. So, having flowering plants, it just looks beautiful. Having cannabis plants and flowering plants, right there. I wouldn't be concerned so much about, the concept that they're competing for nutrients. The plants don't need much. They don't need much to grow, and the fact is that you can grow to any abandoned field. You take a hike, nobody's fertilizing those soils, nobody's watering them. You go to the desert and you got some amazing, spectacular flowers. Nobody's watering them or fertilizing them, and they still manage to get nutrients out of very poor soils or low organic matter. You go to the desert and you see the plant community thriving like nothing is happening, right? And they say, well, this is a very poor soil. Oh yeah, for agriculture, because we tend to feed those plants with gazillion things. And so, and then a plant needs a lot of water. Well, this desert plants are adapted to live on. Nobody waters them ever unless, you know, there rains like once a year. How do they stay green and alive the rest of the year when temperatures are extremely high? You know, they don't need much to survive once they get adapted. And amazingly enough, 95% or higher percent of desert plants are mycorrhizal associates.
That's interesting. I did not know that. You kind of think of the desert as being non-micro, basically the opposite of what you think of when it comes to fungi.
Yeah, and it's amazing what happens below ground. That's why my motto comes. You know, you got to know your friends below ground. The below ground reflects what happens above ground. There's a reflection of what happens below ground. When I work on my dissertation up in the North Cascades on a forefront, on a glacier forefront, the plant community, it was like a mile in length and maybe a 3/4 of a mile in width, and it's just the forefront. It looks like a desert, right? The closer to the glacier, the soil was like very poor, high aluminum, high magnesium, you know, just almost no organic matter, no vegetation. You walk away from the glacier and you're starting to see plants growing on very, very poor soils, but they were growing and they're flowering and completing their cycles. Well, those species did not depend on mycorrhizal fungi. I didn't see any mycorrhizal colonization on those pioneer plants because there's no mycorrhizal fungi on those soils, and the plants are starting to add nutrients to the soil by the roots. But when the roots die, they leave nutrients in the soil and bacteria are starting to grow. So the further from the glacier, the soils are getting older and older. And so we're starting to see the plant community changing as you move away from the glacier, and you're starting to see more species that depend on mycorrhizal fungi, but after the ones that do not depend on mycorrhizal fungi. So
that trips me out. I had the whole picture wrong.
It's a plant succession because there's no propagules closer to the soil. As the plant community develops in age and the soils develop in age and they get more organic matter.
So it's literally the plants driving the system, not the fungi.
Well, it's sort of like a both, because the fungi.
But the plants would start first before the fungi.
Yeah. But and then they're starting to overlap.
Right.
You're starting to overlap. They're still there, but they still don't get mycorrhizal colonization, even though the propagules are there, because those plants, in evolutionary terms, they do not depend on mycorrhizal fungi. They're in the family Saxifragaceae, and the sedges and the rushes, the Cyperaceae, the Juncaceae. Those do not depend on mycorrhizal fungi. They don't give a damn. You know, I grow. I don't care about fungi. I don't know those guys. And as soon as you're starting to move away from the glacier, the organic matter is starting to get developed and the soils are getting deeper. You're starting to see other plants, ericaceous plants, you know, acidic plants, you know, blueberries and heathers and phyllodoces, you know, so those little flowering plants, ericoid, those are fungi completely different than the arbuscular mycorrhizal fungi. Totally different cycle, but they still benefit the plants. And then eventually you're starting to see a lot more arbuscular mycorrhizal plant species, a lot more flowering plants. And then you're starting to see the ectomycorrhizal associates a little bit further when the soils are getting a lot more developed. After 100 years, 60 or 80 years old, the soils, you're starting to see subalpine firs and mountain hemlock, Suga martensiana, and Abies laciocarpa. The trees are starting to show up, but the size of the trees are like really short. I mean, we dated a about 3 feet tall subalpine fir that was 60 years old. 3 feet tall.
Yeah.
Because these guys live under the snow most of the year. And they have very poor soils, but they still manage to be there. They're highly colonized mycorrhizal fungi. So you have non-mycorrhizal, arbuscular mycorrhizal, ericoid mycorrhizal, and ectomycorrhizal plant communities, developing this newly
So
formed.
that's where to go to get the goods. I mean, right basically at tree line. Right.
If you're looking for mycorrhizal fungi that like to live on cold conditions most of the time, because that's the that's the ecosystem. For example, in your case, I would think if you grow on soil.
Yeah, totally.
Okay, so
it's raised beds essentially. I have heated raised beds in a greenhouse.
Yeah, and so do you see any native plants that have been there in your land, on your acreage, that you never plant them? They just show up in themselves. You never care for them, but they're there. They're just green grass or green whatever.
Sure.
They're there. So those are the ones that have been holding the native mycorrhizal population in your land. And so right there are the ones that are adapted to your soils. Those are
so I should harvest the roots and just basically shake them over my beds or put them into my compost.
Yeah, I wouldn't put them in the compost because they will probably they'll get rotten. You just want what I would do practically, you can chop the roots and start your own planting bed just for the purpose of grow a mycorrhizal host on that planting bed. Not for the cannabis, just grow some corn or some grass or whatever, or some flowering, and that holds whatever native thing you have there, and that's what you're going to have, not because of the tops, but because of the bottom, the roots, and basically you're just propagating whatever is there. And again, it is likely that will be happening if you follow the data and then you have evidence. Yes, it's working. Yeah, I brought this bed, didn't have anything because I'm using some kind of a clay sand to use them as a substrate, because that's what they use for commercial inocula. They use really, really sandy substrate because they want all the roots to grow. They don't want hardly any soil in the substrates. They just fertilize with organic, nutrients and let them do their thing. They just water in them and let them do their thing, and that's how they grow them. They don't want to deal with soil because soil brings a lot of stuff.
Extra Trichoderma, all this stuff.
Yeah, all kinds of stuff, and you don't want that. The mycorrhizal system for commercial purposes is rather simple. The less things you have in there, the better, because it's going to help you to harvest it and sell it, because mycorrhizal products don't have soil. They just have that little granular, zeolite or terraphase, green, the little clay, expanded clay. There's all kinds of granulars that they use as a carrier. When I was a student, we had a product that a scientist from Sweden brought. It was awesome. It's expanded clay, and they use it for construction. But it's not angular particles. They're round. They're like little tiny spheres, and they have different sizes. And I still have a little container that I kept 30 years ago, where we used to have the little testing experiments. But the expanded clay is just clay that it was heated at 1,000 degrees, and they just formed these little spheres. And the spheres, the little particles are porous. And so the hyphae and the roots will grow into the spores, and you'll see the spores inside the little holes. You can grab the little sphere full of spores, It was pretty cool to see. And they have, like 2 millimeters, 5 millimeters, and they have even some almost like marbles size, but they use it for construction. And he got it for free. He only paid for shipping. And so it was really expensive because he shipped like 50 bags one time. He paid for the shipping from some Stockholm all the way to Portland somehow. And they got the bags, and we were using those bags like crazy because it was awesome, because it was really clean, easy to work. But that's basically it. I mean, you can use pretty much anything that is clean, no soil. But that's my advice, to get the stuff out of your own land. But again, there's a lot of playing involved.
So something I wanted to touch on is a partial follow-up to something that Josh had touched on, and something that a couple of people have been asking in chat and was on my list of questions. There was a very interesting study recently about, so traditionally brassicas have been taught as non-mycorrhizal plants. But there was a study done and found that with the introduction of Trichoderma, that mycorrhizal fungi actually will colonize many species of brassicas that were typically thought of as colonized by mycorrhizal fungi. Can you talk to us more about the bacteria interaction and how bacteria can first off cause that change in the plant's hosting, but also
I haven't read that paper, and I'd love to see that one. That doesn't, well, I mean, again, there's so many paradigms, there's so many statements that we follow sometimes blind. And I said, and it happened to me a couple of times. We in school we learn, okay, these are the families that are typically non-mycorrhizal species. The family, the Cyperaceae, the Juncaceae, the Saxifragaceae, the Brassicaceae, among others. And so you go with that. And I look at a few roots, I don't want to say a lot, but I look at a few roots, and sometimes they just give them to me. I did a lot of evaluations when I was a student, for many people. I didn't know they were mustard or whatever. They just gave it to me, and I looked at it, and I said, these roots are really clean for some reason. There's nothing on it, not even fungal of other types. And so you solidify that statement. It's like, I haven't seen it. But that doesn't mean it doesn't occur until you actually go after, actually you can inoculate fields of Brassicaceae on purpose to see if actually what, colonized or not. So I would love to see that paper of the Trichoderma thing.
I was going to say,
yeah, go ahead.
On the flip side of the coin, the Forestry Service in California did a study around black mustard garlic, or black garlic mustard, I think it was called, is the common name. I forget the Latin name off the top of my head, but they found that particular brassica actually exudes a allopathy that actually kills local or native terrestrial mycorrhizae, as kind of the opposite of bacteria introduction, but actively trying to kill it off. And is that something else that you've observed in other crops that we should be aware of?
No, I haven't. I don't have information on that. But I was going to say, yeah. So we typically know that there are a couple of things. Well, a couple of things that occur to me that for discussion, or just to throw that in. One question that comes up is why this particular families. Well, the statement that we all know is that 90% of there's two statements. 90% of the plant species in the world, 95, are considered mycorrhizal hosts. Now, it's a misquote, that concept, because Trappy, my advisor, he's the one that generated that statement, but he was misquoted. He says 95% of the plant families in the world have members that are mycorrhizal hosts. So you may have a family, a plant family with, let's say, 100 genera and 1,000 species. And very likely that those genera will be mycorrhizal hosts, but not all of them, because we don't know why. And so each family will have their own representatives. And sometimes all the genera are mycorrhizal. And for example, the Saxifragaceae is another family that we learned that they're not mycorrhizal. Well, I was invited to do a study for the Oregon plant rare species here in Oregon, rare species in Oregon, in the Roseboro area in the central Oregon. So I went with the botanist and says, well, you know, let's go to the rare populations that we know of this plant species. One species was Saxifrag, was a Saxifragaceae family, Benzoniella oregona. Most of this plant was underwater. It was like a little creek, you know. And I said, well, that's the rare species. Yep, that's very small population. I mean, it's like you're talking about 3 square meters, you know, of plant, and that's the only location that they knew about this particular species. So we couldn't collect a lot of roots because we're destroying the only population or the only little plants that we knew. So I said, let me pull some roots out of the water, and I clipped them and put the plant back. I clipped and put the plant back. So we washed them right in the little creek thing. So I brought them to the lab. That particular species had the most beautiful arbuscular mycorrhizal fungi I ever seen. And I use that picture in many of my presentations. So here we go. They told me that Saxifragaceae family is not a mycorrhizal host. Here we are, a rare species with the most beautiful mycorrhizal fungi I've seen. So there are exceptions. You know, there you go. We broke the paradigm, and we haven't been able to publish that thing. I still have a paper to publish. So we got like 14 species with all kinds of mycorrhizal colonization. It's just beautiful thing to see. Another thing is the arbuscular mycorrhizal fungi. It was you're not supposed to see that on conifers, on Douglas fir, on pine, on spruce, on firs. They're not supposed to be there. That's what we learned when I was a student. But when I was a student, I was really inquisitive and always doing assessments and staining. Whatever you give it to me, I'll do the staining and I'll do the evaluation. So one guy brought me 3 or 4 Douglas fir seedlings growing on the greenhouse. They were growing on soil. I said, can you check for ectomycorrhizae on this? So yeah, let me look at it. And when I saw the roots, I said, this roots they look really tender. And a lot of root tips are not ectomycorrhizae because you expected to see a lot of ectomycorrhizal fungi. The root tips are loaded, you know, with the mycorrhizal fungi, not arbuscular. You can't see the arbuscular mycorrhizal fungi. So when I said, let me stain them just to see how they look like. The ectomycorrhizal stained them. Well, I found arbuscular mycorrhizal fungi on Douglas fir. I mean, I create a little bit of chaos because all the scientists and researchers says, no, that's not possible. It's an artifact. No, look at it. Here's the ectomycorrhizal fungi and the arbuscular mycorrhizal fungi growing together in the same root tip. They looked at it and says, oh my goodness, yeah, you're right. And I said, well, what do we make of this? I said, let's go. I'm gonna go and collect more of these ectomycorrhizal fungal hosts, hemlock, Douglas fir, and I'm gonna look. I have an hypothesis. I think the arbuscular mycorrhizal fungi on predominantly ectomycorrhizal hosts like conifers will have arbuscular fungi on the early stages when there's not much competition on the turf soils, on road banks, on areas where the ectos are not right there. They're not in the forest soil. They're not in the jungle, inside. So we collect a bunch of Douglas fir from road banks and roadside, the turf soils. I collect hemlock growing on nursing logs, a nursing log that was above the ground. I can go under the log, and the hemlocks were growing on a row on top of the log, just like you expect on hemlocks. All the hemlocks had arbuscular mycorrhizal fungi, all the little seedlings. All the Douglas fir had arbuscular mycorrhizal fungi. I said, there you go. So we broke another paradigm. We ripped it apart and says, what we thought that these guys cannot have, they can have both. And so after me, I published 2 or 3 papers on that part. And then after me, now people are finding mycorrhizal fungi all over the world in different species, on pine in the Himalayas, in Nepal, and Mexico, and in the US. I mean, now it's well known that arbuscular can be found on conifers seedlings at a younger stage. Eventually they get replaced by ectos. So that example, it tells you how, that's how science evolves. You're breaking paradigms over and over until you actually look into it. Because if everybody keeps following the paradigms that are established, that doesn't mean they're 100% truth, right? And so I just out of curiosity, let's see if it actually, is true. And we found that it wasn't true. And so I feel pretty good about that part, because not always what we think is the truth is correct for every system. So again, I wouldn't be surprised if that particular mustard species is mycorrhizal. And the other part is that maybe evolutionary, those plants do not depend on mycorrhizal fungi. Maybe they're evolved already. They don't need another source of helping them to nourish themselves. They don't need the fungi. They may need something else, but not the mycorrhizal fungi. They don't have the lock and key. It's just like, the concept of lock and key on a pathogen. A pathogen has the key to attack a susceptible host. Genetically, they already know what enzymes, how to break in. And so the ones that are resistant to the disease, they don't have the key. That particular organism doesn't have the key to get in. They don't know how to break in that barrier that is stopping them from getting sick. It's just that's the kind of concept, until you mutate and evolve until you finally, and that's why they keep developing resistant varieties, to stop that from breaking in. But
so with that being said, would it be more beneficial for us to lean towards more lignin heavy cover crops or things like that? It may earlier on, if you're saying that we're finding this level of arbuscular mycorrhizae in those younger trees and bushes, one would think that would be a great way to help it out. Is that something that might be a good idea?
Yeah, there's so many questions and hypotheses that you can follow, trying to understand this, how to evolve the system. For example, I hear a lot about regenerative agriculture. God, there's so many hypotheses you can work on that. It's a great concept, but every system will work differently. Different in Canada, different in Central California, different in the mountains. The IMOs are different, and every little system have to find out what works for you because of the ecotypes, the ecological niche, whatever is adapted to your system, that's what's going to work. So, well, at least higher probabilities, let's put it that way. That doesn't mean the external organisms will not work, but the higher probabilities are the ones that are adapted to your system.
Is it possible? Pardon me for always coughing when I have to start talking. I was wondering this whole time that you were talking about. We talk about oftentimes in cannabis about individual species. So the intradices is, of course, the one that people say is proven, whatever, blah, blah, blah. But you're saying over and over and over again in different ways that the endemically evolved species will do better. So I was wondering if, pardon?
Go ahead.
Well, I was wondering if there's basically a synergistic component to it. So maybe one mycorrhiza is the most effective one at perhaps passing phosphorus or something, but maybe there's a synergy that has to be there so that that species exists. Is there anything to that?
Yep, that's another hypothesis, because the synergism concept, that's when it gets complicated, because we're talking about the simplicity. Okay, you try one species versus another species, and then another block is put them together. You're forcing them to grow together, but you got a third species. Now you got to combine the three of them, in different combinations. So your blocks of experiment design is getting bigger and bigger, because now you have to combine every species with everybody, trying to find what's the combination. What mycorrhizal products do, and a lot of us inoculants, not just fungal inoculants, what they do is that the more you put in there, the higher the chances something is going to work. So you put bacteria, you put mycorrhizal fungi, you don't know which one is it, but one is going to work. And if I put 7, maybe sometimes you hear also, maybe a species A will be good for phosphorus, species B will be good for nitrogen, species C for pathogen, because again, it's a valid hypothesis, because based on the ecological niche, every species, it doesn't matter what, has a function in the ecosystem. Every species has a function. But a lot of times we just don't know what are those functions. We just know that they're good for taking phosphorus, but what else? What else did they do? So every species, no matter what, has a function in the system, and many times we just don't know what it is, or at least.
Bowden stepped away for a moment, but he said before that many species that are considered pathogenic, like Fusarium, oftentimes in a healthy ecosystem actually serve a potentially beneficial purpose. Is that true? It seems logical, because otherwise they would probably be outcompeted from that environment, no?
Yep. And like, for example, the fungi, they got the 3 categories of fungi. Any fungi is based on their nutrition. How do they eat? So you got the decomposers, the mycorrhizal fungi, and the pathogens. That's how they eat, and that's what they do. Organic matter, or they associate with the living plant, causing benefits between both of them, and then the pathogens. They kill the living host. Those 3 things. However, there are mycorrhizal fungi, they know how to decompose things too. They have saprophytic or decomposing capabilities. At the same time, they know how to get into the host and cause benefits. So they know 2 different ways to nourish themselves. Just by the fact that you can grow them in culture, that tells you that they can live on their own without the host. And so, there's so many hypotheses and combinations, and that creates a lot of more fun and a lot of more questions and a lot of answers that we just don't know. So a good experiment starts with the good speculation, and a speculation that is very well formed and educated becomes an hypothesis. And once you set the hypothesis, you can create a nice experiment that will answer that hypothesis, and that's how you're starting to create new information, data. That's the core That's the core of science. Otherwise, we're just making observations. I mean, which is valid. You know, it's nothing wrong. It's just not. It's not direct science.
Speaking of which, another thing that I learned from you and I thought was phenomenally interesting, and I'd love for you to tell us more about, because I'm sure your research has gotten significantly better since the last time we've talked on this topic. You were one of the first people to document aquatic mycorrhizal fungi that would potentially benefit terrestrial plants. And last time I had spoken to you, you were looking into some of that stuff. Is that something that you've done more work on or can tell us more about?
I can't remember what particular. Would that be, maybe that's the one I was talking about, the Benzoniella oregona.
Okay. Sorry for the misunderstanding.
Yeah.
So what are some of the best ways people can promote arbuscular mycorrhizal fungi and endo and ectomycorrhizal fungi that are plant beneficial in their gardens? You do so much awesome work with all these different things. What are some of the best ways that people can utilize the information that you've gotten to help promote the growth of these in their garden?
Yeah, well, what you guys were doing, you know, the regenerative agriculture that's perfect for our promoted, you know, beneficial mycorrhizal fungi. It is very likely that that's what's happening when you do that. You know, the crop rotation has been used by the Aztecs, you know, hundreds of years ago. They call them the milpas, which is basically floating piece of soil with all kinds of combination of plants, you know, corn and legumes and, you know, stuff. And they're all you know, they're just nourishing themselves between each other, creating a little network of stuff. So the more diversity you have, the more chances that you're diversifying your soils to whatever is growing in the ecosystem.
Another question from chat is, will using spent mushroom substrate as soil amendment add any fungal benefit to my soil? It's commonly pushed in the regenerative movement. Are those myco blocks beneficial to my soil and my regenerative garden?
I think so. I think, you know, it depends on how much are you using. I'm just guessing that you don't need that much, because basically those are decomposers, and what you're doing is adding them to break down the hard organic matter, whatever else you have going in your soils. And so eventually those fungi will start to decline once the organic matter is completely, you know, decomposed. It'll just disappear. There's nowhere to go. And the nutrients will be left behind. So I don't know the dosages or the amounts or per, you know, pounds per acre. I don't know, but again, every fungus will do different things. So there's no rule of thumb to say, you know, oysters will do better than, you know, wine caps or you know, other that they sell. So you just do whatever works for you, what is nearby or local. I would start with the local stuff before you try anything. But definitely, yes.
We had another question about amaranth. Is amaranth really as non-mycorrhizal as traditionally preached, or?
Yeah, that's again. So that's what we know. Unless we actually go after all kinds of populations of amaranthus or pigweed, and it's in the Amaranthaceae or Chenopodiaceae. Yeah, it's considered non-mycorrhizal, but again, I wouldn't be surprised.
There's another great question in chat that I really think it's a great question. So Perpetual asks, is there what kind of impact do the various amino acids that people are using in the regenerative side, both organic and inorganic, having on the fungal diversity and fungal growth? Do amino acids have an impact on fungi at all? Are you aware of any interaction?
Yeah, that I don't know. Specifically, there's so many of them. Again, the only way to check that locally is to first identify which amino acids, because there's so many. So it's pretty general to say, yeah, all these amino acids. Well, there's so many. Which ones? I don't know. How would they affect? But I would think amino acid will have a lot of nitrogen in it, and it has to be decomposed for the nitrogen to be available. Is that what they put amino acids in it?
Yeah, so a lot of times that you do it for various different reasons, and there's different ways to isolate them, everything from heat treatments to bacterial isolation to chemical isolation to even some short path and other methods that people use, or fractioning and other things. So again, that's such a diverse question. I just maybe it's too broad. Maybe if it was to ask a specific amino, maybe that's a better question.
Well, I mean, it depends on what the substrate. Where the amino acids come from, and then what would be the high? What type of amino acids will be the ones that are actually more in more abundance on whatever you're applying, whatever substrate or whatever extract.
Is there any organic soil amendments that you would recommend against adding to your soil because it negatively impacts the fungi or mycorrhizae? It's a question I've often heard of, but I've never heard a good answer, and I think you're one of the best people to answer that.
I always question. I always question the high CN ratio substrates. I don't know, but science don't have feelings, but my feelings are that the high CN ratio mulches could be detrimental, because they'll be leaching all kinds of stuff, slowly to the soil. It may be good to protect the plant, but I have a feeling that they could be detrimental to the mycorrhizal fungi in a way. That's my take. The high CN ratio, they're very difficult to decompose.
You mentioned earlier about too much phosphorus being an issue. What parts per million phosphorus level do you really find to be the cutoff? I almost see you laughing and smiling at that question, but this is hotly debated in the organic soil realm. So I'd love an honest answer from an expert.
Yeah, I hear that question. It's the most typical question. The rule of thumb is anything above 50 parts per million can be detrimental. So the between 20 and 30, 20 and 4, between 20 and 50, apparently it's a good point for mycorrhizal colonization. 20 and 50 parts per million.
So maybe as a follow-up to that, what is the optimal N to P nitrogen to let me say this better. What is the optimal nitrogen to phosphorus ratio for fungal growth? Because I know what it is for plant growth, but I've never heard an answer about what it is for fungal growth.
Yeah, I think I mean, it will again, too much nitrogen also in the paradigm is that too much nitrogen, too much phosphorus is detrimental for mycorrhizal growth, colonization. And the organic fertilizers that the commercial growers use usually is about 888.
Is there any minerals or ratios that might offset those particular numbers? So say if you had a certain micronutrient or maybe or macronutrient, or is there any micro macronutrients maybe also aside from phosphorus might be inhibitors of fungal growth?
Not that I know, but pretty much anything in excess is bad.
I've also heard sulfur as well. I've heard of secondarily being, at least in excess levels, being fungal.
Yeah, in a very, for example, this in the case of ectomycorrhizal fungi, the French black Perigord truffle, the French black Perigord black truffle, they like 7.9 pH in the soil. 7.9. That's the optimum pH for that particular fungus. And so on orchards outside France and Spain and Italy, they have to bring their soils higher by adding calcium carbonate to bring the pH 7.9, because most of the soils where you're trying to put the truffle farms are acidic. And so they have to really heavy on the calcium carbonate to bring the 7.9. So it tells you that every fungi will have their own little thing, their own little recipe. So it's very difficult to generalize what type of minerals is detrimental. It really depends on the species. So it's hard to generalize.
Will the fungi kind of unlock, how do I say this? Because the soil fungi you said are colonized by a myriad of different spores. I assume there's the ecto and endomycorrhiza in the soil, just in all kinds of different species. Will they basically solubilize nutrients at different pHs as the soil swings up and down through the season?
Yeah, basically, well, as on many soil systems, the enzyme production by all the soil organisms, all the decomposers, all the mycorrhizal fungi, the enzyme production, the types of enzymes is what moves the dynamic of the soil. Without those enzymes produced by those organisms, you can't break down all those compounds that are not degraded, because the plants in all the soil food web of organisms, they can absorb all those complex compounds. They need to break them down to the minimal size so they can absorb them. And so, because they need to produce extracellular enzymes out of their body to break down those components so they can absorb them from the outside in. It's not like us that we have a mouth and you have to chew it, and the extraction is in the gut. This guy have to do it outside because I need to get nutrition from the outside in. And so the enzyme production, it's a jungle out there. I just can't imagine a computer machine to detect all the enzymes that are being produced. Well, usually it's going to be driven by what's the substrate that is in higher amounts here that needs to be breaking down. And so, you have a lot of leaves. Well, there should be a lot of organisms trying to break down those leaves. And to break down those leaves, we need enzymes that will break those compounds on the leaves. Then you get a piece of twig and all kinds of different types of organic matter at different decomposition levels. So the enzymes keep changing. So it's very complex, and we're always trying to understand a very complex system with very few little words. And as I said before, if you try to put a diagram of arrows of what's happening in the soil, it could be millions of arrows going everywhere. What affects what. So
with that said, if everything's interacting constantly and sort of recycling everything else's waste and on and on and on, is there ever an overdose of fungi in the soil, or for that matter, even maybe bacteria? Like, do you ever have too much?
I think when you say overdose, I'm thinking that there's so much of one substrate that one thing becomes the dominant. It could be, but that's the concept of, for example, when you have too many of one species and that species happen to be a pathogen, well, it's going to be disease. A disease happens when there's so much of one particular susceptible tissue And the population of this pathogen is just overwhelmed. So like there's a lot of food here, and it becomes a disease on your field. That like, darn it, I got blasted by this. Well, because you put a monocrop, and it's just one single thing, one pathogen find the habitat, and it's just the rest of the field is the same food, and we know how to break it. So the humidity is fine, the temperature is fine. We like it here. We're just gonna grow, and they wipe out your crop.
We had a great question from chat. How do you measure or quantify in your home garden as a maybe less experienced grower whether or not you have healthy root fungi growing on in your system, and whether or not the fungi in your system is actually beneficial? That's a great question. It's something I think a lot of people just have no idea.
Yeah, unfortunately, you can't. You can't measure unless you look at it under the microscope. There's no other way. You can guess. I mean, as I said, if you put mycorrhizal host plants that we know for sure are gonna propagate the below ground ecosystem, it is likely you're gonna have a mycorrhizal community. But we just don't know how many. There's no way. They're microscopic organisms. You can't see them. You can't see them with the naked eye. They're transparent. They need to clear the roots and stain the roots.
Is there any visual way on the plant that you could tell if you didn't have them or anything like that? Okay.
No.
Yeah. Are there specific mycorrhizal fungi that are better for container growing, or maybe grows that don't directly connect to the earth versus outdoor?
No, nobody knows.
Val actually asks a good question. She has a microscope, so she says, "How?" I admit this is probably an intrepid question, but she's quite intrepid. So, how do you actually start learning about mycorrhiza with the microscope?
Yeah, so the method that I use has been modified for the years, but it was intended. It was developed by a plant pathology guy working with nematodes, and he wanted to check out the nematodes damage on the roots of a citrus trees. And so he cleared the roots with potassium hydroxide solution, 10%, and heated up in a solution, the roots, and then the KOH, as the potassium hydroxide, clears the pigments of the roots. So all the color, all the dark stuff that you're trying to get rid of it, so you can see what's inside, it's done with the potassium hydroxide. And then you neutralize it with hydrochloric acid solution to neutralize the KOH in the roots, and also acidifies the root cells for the stain, because the stain is an acid base. And so the stain goes inside and makes its work, stains the root cells. And so you have to clear them, and then neutralize them, and then stain them. And then I use Trypan Blue at a 0.5%, which is like half a gram on 1 liter of solution of lactic acid and glycerol in water. And then you'll be able to see the root damage by the nematodes. Actually, the nematodes damages and the eggs look pinkish, which is very helpful. But he noticed that they stain the mycorrhizal fungi. They happen to be inside there. So that's how the method got developed by some nematologists. And so, we use that method because their fungal hyphae is microscopic to begin with, 5, 10 microns in width filaments. And they produce vesicles inside the roots, and they produce arbuscules inside the roots. So these structures are transparent. And if they're transparent and they're inside a pigmented root, you can't see them. And so you stain the fungal hyphae with this stain, trypan blue. There's other stains, but that's the one I use.
This is a common misconception, isn't it? At least among cannabis growers, you always hear this, especially among like no-till growers. We'll put down some barley or honestly any good organic-based amendment, and the next day there's some fungus on the top or some pseudo fungus, and people say, "Oh my God, it's mycorrhizal." That's not it at all, is it?
They're decomposers. Yeah, mycorrhizal fungi, you can't see them. The spores, they take their time to germinate. It could take 2 or 3 months for them to get in. And so that's another challenge for cannabis growers, because if mycorrhizal colonization doesn't start earlier, there's no much point because the cycle is rather short. And so you want to start the mycorrhizal colonization, you want to start it earlier. You put the inoculant way earlier and your substrate on your pots way earlier. So they're set and steady, and they're just waiting to be waking up. They're just waiting in the substrate. Because other people put them at the same time, you put the plant, well, the roots are going to grow and the spores are still sleeping. You put 1 month, 2 months, and the spores is, okay, let me germinate. And the plant is already done. And the fungus says, what's happening? I'm just waking up. What's going on?
The American one actually asked a relevant question. He said, are there specific mycorrhizal fungi that are better for container plants? I mean, you're saying that actually plant them earlier. Can some of these, I mean, many people have well, at least I've been taught, frankly, that the mycorrhiza don't survive when there's not a plant root that's alive. How would you plant the fungi to start them ahead of time?
Because what you're putting inside the spores, their spores are naturally dormant. They're resistant spores. They have thick wall. They need to be sit and still for a while. And then they need to be stimulated. Some people put them in the fridge to cold treatment to wake them up.
So I've heard of people putting their bags of mycorrhiza in the freezer, and I just thought that was stupid and just some kind of a bro science, but that's to actually stimulate them. Yeah,
they stimulate. If you think of nature, that's what happens in colder parts. The natives, they get frozen. The soil, the native mycorrhizae, and the winter, they go under ice, snow, and what happens in the spring? They come out, the plants nicely. Nobody inoculate them; they're just there. The natural cycles include cold temperatures in this part of the world, and so if you have isolates from the tropics and you're trying to use them in the northern hemisphere, they're just not adapted. They may work, but they don't know the system because they're not genetically, they're not adapted. You gotta work with what you have around.
So when would you recommend introducing mycorrhizae? At the point of germination or short, like a week in, 2 weeks in, or?
As soon as you get the soils ready, if you use pots, get your pots ready way in advance. Put the inoculant in the freezer or in the fridge way in advance, and then put it in your substrates. And then you let them sit there. And then once the germination happens, the spores are already, they've been sleeping for I don't know how long, the more the better. They're not going to wake up. They take a while.
Maybe on the other end of the growth cycle, is there anything that growers can do to kind of encourage spore production at the end of a growth cycle?
Well, if they don't, no, it's just let the plant do whatever they're doing. I mean, what happened? I think they're doing it already because, like for the cannabis, it's a natural thing, and it's an hypothesis because cannabis has been modified for so long. We're assuming that it's still physiologically are still doing things the way they're supposed to be when they were rural, when they were wild in the wild. We're assuming that. It's very modified crop now. So we don't know if the physiology, at least I don't, the physiology is still recognized by the native microorganisms. See, I don't know. So because we're using a very modified plant, and so we're just assuming. I mean, unless it's tested. But the cycle of the cannabis, it's once starting to produce the flower, the physiology of the plant is changing. You know, the leaves are changing, the roots are changing, and so that sends a signal to the below ground community that my host is dying. If that individual is colonized, if the host is dying, the mycorrhizal fungi senses, quote unquote, that I need to produce spores because my host is going to die. I need to propagate myself, and so they produce spores. In theory, that's what they do in commercial production. They stress the plants so they can produce spores.
That's super interesting. I've never heard about that before. That's really cool.
And
Well,
so stressing the plants, you know, it causes chaos below ground, right? It changes the whole thing. And say, hey, the host is dying. What are we going to do? We depend on this guy.
So, I mean, in theory, are you creating some kind of a chain reaction? Like we were just talking about this yesterday on my show about how, and many times on Potent's show, I think on this weekend, that stress basically in different ways causes plants to increase terpenes, you know, stuff that we're interested in. But then at the very end of the cycle, if you're already stressing the plant in some way, I'm sure there's, 50 different ways to do it. You could be potentially, if let's say you're in a no-till environment, you're like in an organic situation, or maybe outdoors where you're going to reuse the soil next year. Are you then also stimulating the mycorrhiza to create spores for the next season?
It is likely. It is very likely. As again, I have to be careful. I'm not.
Sure, you can't say for
I don't
sure,
hold the
but
truth, but it's likely, and the hypothesis leans that way. So unless it's tested, again, that's what science. Because I'm always thinking that science peers always listening to me, and so I gotta be careful what I'm saying, because if I say something that is out of the science Bible, I'm gonna get criticized. So you have to be careful on the statements. I don't hold the truth. There's a lot of hypotheses, the likely, the probability is the potential, all those words that are key words to for liability issues. So you learn that in graduate school, like you gotta watch what I said, because I don't hold the truth.
This is a great question we got from chat. Will transplanting into an already used, freshly harvested, inoculated root ball from a previously growing cannabis plant speed up the process of the next plant being inoculated with spores as those things are dying off? And this is, maybe also secondarily would go along with having those cover crops as well, being part of that.
Assuming that those roots are colonized heavily, preferably, and then yes. But you have to be careful. For example, if you're going to leave the root ball as it is, how that root ball is going to decompose? You gotta help. You gotta help out. Help that root ball. Grind it or fragment it or chop it. But again, it's gonna be a lot of work. If you have hundreds or thousands of plants, how are you gonna do that? Well, that's something that is out of my league. But I can fantasize or make a recommendation to say, hey, get a grinder, get a big grinder. We're assuming that those roots are colonized, because if they're not colonized, well, there's not going to be much mycorrhizal fungi there. There are going to be other things that might be helpful, but if you grind those things, especially dry, not fresh, dry, the fungus will still viable even if it's dry. And then use those fragments and place them in the soil. That in theory will be, pre-inoculating your things, assuming that those roots are mycorrhizal. If you don't know, if you don't actually look at them under the microscope and have an evaluation to say, yes, I bet the 50% of the plants in your field are mycorrhizal, and then you have 50% colonization. So you mix them all up, you grind them all up, and then you're gonna have mycorrhizal propagules in your system, assuming that they're mycorrhizal. If you don't know, and then you don't know, you're just grinding the roots. Whatever is there, right? So,
and so you would have no way of knowing.
You have to.
Is there any way of knowing? Everybody that's been listening has been wondering. Is there any way of looking at the plants and knowing that they're mycorrhizally associated? No,
because I've seen some comments. They pull the roots and it's all hairy and white and beautiful. Look all this stuff and look at the mycorrhizae. It's like, oh my God, they can't see them. They're root hairs. And if there's mycelia, that mycelia is not mycorrhizal fungi or decomposers. They don't form those white mats. They don't. They're transparent. You can't see them. If you're talking about ectomycorrhizal fungi, that's a different story. Those grow on pines and firs and Douglas firs and spruces and oaks. Those actually form mycorrhizal rhizomores, like strands of hyphae all bundled together. But those are different fungi than the ones that Cannabis use. They're totally different fungi. Those are different. Those are ectomycorrhizal. Those root tips, you can actually see it with the hand lens. The mycorrhizal root tips, you can see them.
Those are the ones like when you're in the forest, for example, and you pull up an
Yeah.
old log or something, you see all the
see the root
stuff.
tips.
That's the one.
Most of those root tips, they have different colors and things. Those you can see with the hand lens.
Do both of them fruit, both ecto and endo? I mean, probably not every species fruits, but do versions of both species fruit?
Yeah, well, see, the arbuscular mycorrhizal fungi, the ones that grow with cannabis, do not form mushroom. The only thing they form is spores in the soil. That's it. That's their cycle. They can't see them above ground. They don't form mushroom. That's another thing to clarify. They're totally different. Spores, hyphae, root system, spores in the soil. They're left behind, period. Some are developed inside. The ectomycorrhizal fungi is a totally different fungi, total different cycle. The spores are very small, 5, 10, 15, 20, 50 microns. They form the hyphae, they bring the colonization on the root tips, they form a structure, they form a mantle, they change the morphology of the root tip. And then they make the strands and mycelia and mats all over the place. Those can be confused with decomposers. They can be confused.
And so that's why to kind of clarify the point, that's why you grind up the roots, basically the mycorrhizally associated plants, because that's the only way that you would potentially propagate the spores, correct?
For the arbuscular mycorrhizal. Right. Yeah.
As opposed to you could just shake a mushroom or something for the ectomycorrhizal.
See, the ectomycorrhizal, they actually can form a mushroom or a truffle below ground, depends on what species you have. And so the spores are dispersed by air. It comes a mushroom, air. And if it is a truffle, they're dispersed by small mammals. They eat them and they spread them. They go and poop somewhere else. And so that's how they spread. The mycophagy is called. So the concept is called mycophagy, fungus consumption. That's done for the truffles and for the mushrooms too, but the mushrooms are dispersed by air, spores. They produce millions and millions of spores, millions. And the arbuscular mycorrhizal fungi, they don't produce millions, they produce hundreds of spores, not millions. And so they're very different cycles.
I've wondered why bags of mycorrhiza always have so few, and the spore counts are so tiny. And
it's called business. And also, well, it's a business, and also their system. They that's their business. You know, they mycorrhizal fungus per production is very difficult to control how many spores you put in a bag. Not all bags are created equal. You assume that they're uniform, but not always. So it's very, very difficult to make a homogeneous product. And so that's why there's so much competition.
One thing down that realm, something I really wanted to ask you before we wrap up the show here soon. Mycorrhizal color. We've had quite a few questions about this in chat. We're often told that white fungi, fungal strands under a microscope are beneficial, and the non-white ones are not. Is that true? Because I've heard quite a bit of evidence against that, and I think you're the best.
It's one of those statements that are really general. It's not. I mean, there are pathogens that can form white strands. There are decomposers that can form white strands. There are ectomycorrhizal that can form white strands. It all depends on what you're looking at. You have to, put more factors in to make a statement at a particular thing. No, the diameter of the hyphae also, I heard that statement so many times. The diameter of the hyphae doesn't tell you if it is a good fungal or a pathogen. Nope. I mean, I cringe when I hear that, because you're talking about 100,000 species, and the diameter of the hyphae is all over the place. It doesn't tell you if it is a saprophytic or a pathogen or a mycorrhizal. That doesn't define what they do. The diameter, no. And the same thing with the spores. There are pathogen spores that are small, and there are beneficial fungi that are small. So to actually know that one is a pathogen is based on experience and science. Like, for example, Armillaria melia. It's a fungal pathogen, the honey mushroom. It's a root pathogen, attacks the trees. It's an edible and produces strings that look like shoelaces. They call them shoestrings. They're black rhizomes on the bark of the tree, and it produces this mushroom. But it's a beautiful mushroom, but it's a pathogen. It produces white mycelia again. There's they call them white. What is it called? The brown rotters and white rotters. It depends on what kind of sugar they're attacking, right? If they eat the cellulose, they leave the brown wood behind, right? And if they eat the brown, they leave the white wood behind. So there's some fungi that decompose the lignin or favor the lignin, and some fungi favor the cellulose. And so there's so many statements out there that they're just confuse people or give the wrong statements. There's so many things out there. There are no rules. How are we doing with time? My goodness, you told me 1 hour is 2 hours.
Interesting, that's for sure.
Apologize for running long there, brother. We'll wrap up here for you. We do love having you on.
I'm missing my cookies and milk, man.
I'm sorry, man. Here, we'll let you know, tell everybody one last time how to find you. I'm going to throw your website up here again.
Yeah, you can find me on MycoRoots, simple, or on Instagram. The friend that I was in Central Park doing some work in New York, and I wasn't on Instagram, and I said, could you make me an Instagram account? I had no idea how to do this. And I give her my phone, and she says, here, you have an Instagram account now. And then I forgot about it. And months later, I said, do I have an Instagram? What's the password? I had no idea what the password is. So I'm afraid to change phone because I may lose it. So I don't know why MycoRoots 1258 came up. I had no idea what that number means. That's what it means, I guess. Maybe it was taken already. There's a company in South Africa. It's called MycoRoot without the S, but they produce inoculant. And they called me like, I don't know, 15 years ago. They sent me an email that says, hey, that name is mine. I said, no, I think I got it mine first. So they stopped bugging me. But there is a singular MycoRoot, and they produce inoculant and they produce inoculant in South Africa. I'm showing off my my t-shirt.
Awesome.
Anyway, so yeah, feel free to add me or whatever.
Awesome.
With
What type of things can people reach out to you for if they're running into issues in their garden?
we'll send them an email, MycoRoots@comcast.net.
Awesome. Well, I very much appreciate you coming on and sharing your knowledge. You're truly one of our favorite guests to have on the show, and I thank you again for coming on and talking to us about all the wonderful work that you're doing.
All right. Well, thank you, Steve, and hopefully we'll see you sometime in one of those meetings. Maybe in Oklahoma. I don't know what you're doing in Oklahoma, man, but
Sure. Oh yeah. Hey man, if you had told me 3 years ago I'd be living in Oklahoma, I would have been like, you're crazy.
well, that's how long ago I saw you in Portland.
Oh yeah. Yeah, that's right. Last time I saw you in person was at the Portland Regenerative Conference. That's right.
At the McMiniman bar.
Yep.
Good
times, man. I think it was you, me, and Dr. Faust, and we talked well into the night on all kinds of stuff.
I know, yeah. Josh was also a troublemaker.
Josh, yep. All right. Well, thank you so much for
all right. Well,
very much. I'm grateful for your time tonight.
Okay. Good luck on your classes and your podcast, and have fun, man. Let me know if you need any help.
Sure. Appreciate it, man. Thank you.
See you. Good night, everybody.
Take
Bye.
it easy.
No kisses for you guys. No. How do I hang up? I have no idea.
Just hit end there on the call.
On the little
whatever.
On the little cross.
Yep.
Okay. All right, man. Bye.
Thank you.
Just throw your phone at the wall. And if it doesn't work the first time.
Bye, Fumidor.
Cheers.
Oh, that was another awesome episode with Dr. Efron. He is such, but if you haven't checked out the first time he was on, he dropped an equally deep amount of knowledge on the show. So I very much appreciate him coming on. He is such a good guest, and I certainly learned a whole lot tonight. We also have Brendan here. You want to say hi?
What's up, Brendan?
Hi.
How's it going, Brendan?
Do you want to tell everybody maybe a little bit of what you've been up to or what's going on over at SK?
Other than getting bumped off a flight and having to fly around the whole country to get home a couple days ago. Yeah, still doing obviously Spectrum King LED, Biocat Solutions more. And I've just been accosted by a savage cat named Killer who's decided to get on the show now.
Oh, yes.
Yeah.
Of course. He was the one meowing.
They're related to lions, they say.
This particular one he holds his own with 2 pits and 2 wolf dogs. So
yeah, he's the boss.
He really is.
He's the boss, and the dogs leave him alone.
Oh yeah, for sure.
There you go. But yeah, I do the same stuff. Obviously, I'm still working on finagling and making a new system with Steve on one thing. I got the Biocat Solutions more product, which we're excited. It's more commercial growers that are sciencey, dare I say, who want to get under the hood with it, become fanatics with it, because that's, in fact, I was really digging listening to Dr. Efren Cazares speak, because I was thinking I would love for somebody with his level of knowledge to play with this product and some other ones out there and give his feedback on what he thinks that he sees them doing beneficial for a different swath of plants, for example. That'd be pretty cool. I'm just, at the end of the day, obsessed to finding a way to grow the cleanest, healthiest, safest wheat. Whether it's lights, nutrient, something else, it's kind of what keeps going around in my head.
Yeah, and I've definitely used the more product in quite a few aquaponic trials now, and It certainly helps keeps the roots cleaner and reduce breakdown of sludge in
Keeps
the
your
system.
fish cleaner.
Yeah, it's a good complement to stuff like labs. It's a completely different species, group of microbes for your system. So that complements labs well. But it's certainly something that, like, again, through trials and testing it, I like it a lot. Even if he wasn't my friend, I'd be using it. So I just happen to get it some samples before everyone else does. That's all.
He knows a guy.
Yeah, I know a guy about a thing.
Yeah.
And then what else you been up to? You were just off to Vegas. So how was the
Oh,
conference?
the MJ BizCon. That was a month ago already. Yeah. That was interesting because I honestly didn't think there'd be that many people there this year, but I think they said 27,000 or so showed up, and it sure as hell seemed like it. It was a very busy show. What I saw clearly is there's still what I call the tire kicking public, where people who think they're going to make a ton of money in cannabis who've never done it before, and they're coming around checking things out, trying to get a feel for it. Then you have these commercial grows who come in who are now just absolutely huge. Commercial grow-ups. I mean, 10 years ago, there was a handful on Earth. Now there's, millions of square feet here, millions of square feet there, overseas, you name it. Now, a couple more countries are going legal. And now I just found out that Brazil might go legal. So if Brazil, somebody I know said there's a bill in the docket this year, it probably won't pass, but it'll pass next year.
Just today, there was a story about Germany. Evidently, Germany is pushing forward.
Germany intends to put it through. Yeah, I saw that news. I mean, so if I had thought, 15, 20 years ago, this wouldn't have entered my mind as a remote possibility, that this many people would catch the benefit. What I'm sad to see is like states like California who now intend to raise taxes on something that, they're going to reintroduce the black market in other ways because they're taxing people out. It's a commodity like anything else these days, but it shouldn't be taxed discriminatorily like it is in most places. That's something I see from all the places I get to. Some countries, what's the word I'm looking for? They finance under the hood. They create benefits and limits on what growers can make on the product. So only conventional low quality producers are going to get in because they have the best chance of making money on it, even if it's small numbers.
I remember the legalization, or at least the I think medical program anyway in Poland was so draconianly shitty. They import the cannabis from, I think, Switzerland, or I don't even remember exactly which country it is, but anyway, they import it from a Western European country that's actually pretty expensive. That's why I think it actually is Switzerland. And then they import it pre-bagged, pre-ground, pre-something else, then it's irradiated on and on. And the prices basically would be the equivalent for a Polish person once it comes to their dispensary or whatever, once it comes to their pharmacy, would be like $100 a gram, for something. I mean, just the shit.
Exactly. How to guarantee people won't buy it, right there. If you could get a seed,
the swaggiest weed you could find.
Yeah, but if I'm a home grower, or I'm somebody who wants to now grow at home because I can't afford it, but I still want it, there's your black market birth. Well, I'll get it to you for 50. When really it should be, what the local market can bear. I want $100 to a Polish person. It's probably like $200 to $500 here.
I mean, that's what I'm saying, like the equivalent, for a freaking gram. I don't even remember. It might have been honestly been more than that, but it was for a very swaggy system that nobody was going to engage in.
And there was one of the South American countries that did that. And
Colombia?
No, it wasn't Ecuador.
No.
They made it so the rules are so weird that.
Hold on, I know what you're thinking of, because my buddy, I think, is the only legal operator there. What is it called? It's in northern.
I'm not going to remember the name.
Pull the map up. Hold on.
But
Joshua Steensland is part of that group.
They made it, so difficult in that particular location for people to even get permitted to get the cannabis. Then they can only go to a specific pharmacy, and they always made it so it was like hours away from them.
No. This is different. Suriname only has a handful of licenses, and one of them I know Joshua Steensland's working with.
Okay.
That's the one I was trying to remember. But was it
Yeah,
Guyana
so
that you're
that's just
thinking of?
another, well, it's just another example of some place trying to slow it down by making the rules lugubrious, we'll go with.
I know there was one of the Suriname was interested only in people that were not going to come in with like heavy chemicals and stuff, and that was
Mhm.
one of the reasons why you see a lot more regenerative people moving into that group. But it is interesting to see how the different countries regulate that in terms of what they can and can't use. Jamaica has some pretty strict rules on what you can and can't
Mhm.
use, and some of the other places. So it's nice to see at least some of the patient protections being passed on to some of these places that maybe you don't think of as being as
Yeah,
progressive on testing.
actually, Macedonia, another example of, it kind of got goofy there, but they developed a healthy market for it, you know?
Hmm.
Yeah. So,
If
there's a
you
lot in a lot
speak
of
Greek, go
countries.
to Macedonia. Don't they speak Greek there?
I'm
Maybe.
not familiar with which language crosses what line. It does cross the line into Macedonia because it was considered part of Greece at one point, but they have their own language, and I think their language is a little closer to the Slavic family of languages than Greek.
Those of you that don't know where that is, north of Greece.
That's
There you
where
go, man.
Alexander the Great was from, I think.
Yep.
It's between Albania and Bulgaria.
Yep.
All those countries, it's all Slavic languages. They're pretty wild. They change quite a bit.
Cool.
But what I was getting at earlier is that with all this legalization going on in so many places, at some point, whether we like it or not, there's going to be like the Coors Light, the Marlboro of weed, you know, and it's just going to be the countries that can produce, you know, any Ecuadorian nation that can produce year round is going to win because their price per production is going to be like in pennies at that point, and they're going to sell stuff at 1,000% margin to them, and it'll still only be $10 a gram here, unless governments continue to tax insanely, which they seem to think is a free money flow to solve all their corrupt problems.
Is this true? Somebody in chat just said that the current price of New York medical dispensaries at the moment is $55 for a half gram of
Oh, wow.
ground
So
up
when I was saying $100 for a gram, like that was just people were like, "Oh, that's not bad."
Well, again, that just shows another example of a state taxing things
Right.
the wrong way. And I'm originally from
But
New York.
that's fucking crazy because Japan, allegedly, the black market rate in Japan is $80 a gram.
And that's where like it's like a death penalty, isn't it? Like it's pretty severe to get caught.
So, yeah, it's crazy bad if you get caught with it. How the hell is that higher in a legal tax rate than Japan? And they wonder why they still have a black market. when you can buy pounds in Oklahoma for $250, $300.
It's dumb.
Well, I want you to, granted, you could buy pounds of that, but would you with it?
Well, it's
Smoking it,
garbage
not too much.
outdoor, but I'm just saying is that if you're paying $55 for
It's-
half a gram, that's you happily buy a
Okay,
B
for that
grade
one,
for that.
I'll go with beef fuddling.
Fuck. That's better than any street price that I've ever heard of on the East. I grew up in Philly and we never ever heard of anything. Even the highest I think I ever heard of anyone actually paying was maybe 5K.
I mean,
4 or 5
110 bucks for a gram, that would have to be the best gram in all of the city. Like, honestly, like it glows in the dark and it's fucking gorgeous and makes you see stars and I don't even know what makes you high for 3 hours, 10
But that's-
hours.
I'm sorry. Here's a better question. If you have cancer or some other ailment, you're
Kind of
looking
fizzle
at like, how would you afford? You're looking at like 20 grand a month.
Yeah, you better be a rich CEO or something.
But that's fucked. Like what?
No, that just scares.
It's dumb. Oh, you guys are muting yourselves. Brendan's saying really interesting stuff, but it's muted.
There's someone else in chat saying they've also seen things for like 55 for an eighth. Even at 55 and eighth, that's still fucking crazy. You can get
55 is, I mean, that's about what well, it's cheaper than that now, but it's what it used to be about 5-6 years ago before I started to really grow. Like, That's actually honestly if like 40, 50 bucks for premium eights. So 55 is already on the kind of the higher end of that, but like really nice eights, you know, that would be something like really flavorful, really like super purple, you know.
Yeah, but we both know they ain't fucking primo in this dispo that early
That
on.
was the dispo version. Yeah. I mean, you get spoiled pretty quick when you, that's the thing. Like,
Well,
when's the last time I bought something at a dispensary? I mean, every once in a while I'd go, but I was always disappointed. I was like, really?
That's a great question. So, Fumi, why don't you tell us a little bit about and we all three of us know the difference day and night, but about how different homegrown cannabis is to you know, commercial brought. Yeah, because in commercial settings, people don't realize it's all about turning, you know, the maximum yield as fast as possible, and everything else be damned, really, outside of a few, you know, side companies. But most of the time, they're going for the largest bud producers that are the most mold resistant. And that are the fastest turn.
I mean, in an operation, how do I say this, in a commercial operation, you wouldn't ever think about going over your deadline. First of all, because you don't want to. It's not your pocketbook. Second of all, your boss would freak the fuck out because he's, you know, trying to churn it and blah 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 blah. So you keep to the schedule, and that schedule is perpetually, as we always hear, and you can see it if you know anything about weed. It's perpetually underripe weed, right? How do I say this? It's not matured, you know, especially some of these sativa strains. People always say, "Where's all the giggly weed?" Da da da. They don't grow it anymore. They don't want something that finishes in 11 or 12 or 13 or 14 weeks. They want it 7 weeks, 8 weeks, and done. Whereas consistently, one of the things that I notice with home growers, I see it with myself, they baby their weed, and they will always, almost everybody, give it the little bit of extra time that it needs. And that could be 1 day, that could be 2 days. And with weed, it could be 1 day, it could be another week, could be 1 day. They'll give it that time to let it get greasy and delicious and delightful, and they'll basically baby it until harvest, and then they'll cut it down gently, and then they won't beat the shit out of it and leave it in an empty, you know, room dry and leave it there for 3 weeks. Like, they'll basically take care of it after they cut it down well as well. So just this complete, how do I say this, this combination of effects, basically. The fact that they jar it up lovingly instead of throwing it in a bale somewhere and then just forgetting about it, and then they don't let it sit for 6 months. You know, this is actually one of the biggest issues with dispensary weed. I believe that there actually is. I've seen some pretty decent weed from commercial operations and so on, but then by the time it gets to the dispensary, a little bit swaggy because it's been sitting around for 6 months. You know, it had to go tested, and the sample that went and gotten tested was, you know, blah blah blah, but the other sample was just sitting somewhere. Maybe it was climate controlled, maybe it wasn't. Whereas with the grower, it's always at hand. It's usually smaller amounts. They smoke through all of it, and then the next harvest comes. You know, a lot of times, very common thing with growers is they run out of the previous harvest and they're waiting apprehensively for the new one. So there's always fresh, sticky, greasy, terpy weed in their stash. You know what I mean? And the dispensary almost never has anything like that.
Muted, right? That's why.
No, you're good.
But, you know, knowing when to harvest means you have to know your plants and monitor them the right way. You know, what Steve mentioned is true. I've been in some massive commercial facilities that I've sold lights and things to, and I watch them and they're like, it's chop day. I'm like, these plants aren't done. The grower's like, I know, but they told me it's chop day. So yeah, you know, they're taking stuff which sometimes is 7, 10,
even if it needed 1 more day. Too bad.
Yeah,
no. It's period. It's just chop day. You said 57 days. Tomorrow's 57 day. I'm like, you know, but if the weather was different this season, this last season, you know, like if you're in a greenhouse, they may have done it in 57 days because it was hotter, you know. But now it's a few degrees cooler, and that plant needs another 3 days, or maybe even a week, or maybe even 10 days. Worst case, it's not going to get it. So you're getting weed that wasn't properly flushed, that wasn't grown to the sweet spot. You know,
and I mean, this isn't a joke either, because the commercial operations will bring in a crew to basically harvest and, you know, later on trim that greenhouse. They don't, well, I mean, some places probably are big enough maybe to have a full-time staff that, you know, rotates from job to job to job. But I hear over and over again about harvest crews that basically all they do is go from site to site to site, and that's what they do. They just harvest a crop. And so if you have a contracted harvest crew coming, they're not going to wait for 3 days, because that's the next job for them. You know, they're already moving on to something else. So they're there for your 3 or 4 or 5 days or whatever it is, a week or something. And if you basically let them go, they're going to be somewhere else. Go ahead.
You know, the problem is the size of your grow and the training of the staff. I mean, if you've trimmed your own, I've trimmed, you know, it's an art form in and of itself. And if you have just a bunch of hackers you don't know coming in and whacking away to get, because they're getting paid by the perceived pound hacked, and I say hacked, not trimmed, because that's what it looks like to me a lot of times.
True.
That's degrading your value as well. So if you're going, not running your plants to true fruition, and you're pushing them out and pushing it through, it's kind of from front to back. This plant had a bad start. It's not going to have a great finish, and it's not going to be the quality that someone that's grown or had someone like you mentioned earlier who really takes care of their plants, who's really monitoring everything and gets them right at that point. You know, when you see some people want to see 20, 30% amber, some people want to see 10%. That's personal taste at that point, but it's within an acceptable zone. But if you're chopping the plant where the trichomes haven't even turned milky yet, you're getting a totally different flavor profile out of that animal, a totally different effect, because it hasn't hit the sweet spot. And I know that may be redundant, but when you've had plants that have been in the right spot, there's a different effect on you. There's a different flavor, different palate profile. All of it's different. If you've had harsh, evil, you know, a lot of times that's because it was cut early and it wasn't flushed properly.
And that's
That was one of the things. Go ahead.
That's something I've often butted heads with owners and managers on it as a consultant, is basically trying to teach them when is actually harvest time, and that's when it matters for turnover. And you know what, your veg might be bigger than you thought it was going to be, and you're going to have to prune them a little bit heavier. But you know what, tough titties. Like, you wait, you know, you screwed up your schedule, you didn't know your cuts, and you didn't know how to speed them up a little bit. You know, if you jack the temperature of the room up a little bit, you can speed them up. In fact, Breeder Steve was just talking about this on Sunday. It was one of the first talks. He was talking about nighttime temperature being elevated, being a huge factor in speeding up senescence, and how the root structure, the root temperatures, and the foliar temperatures at nighttime being a huge factor in senescence and accelerating senescence.
Val here had a great point, by the way, about Frenchie being a big believer that basically everybody chopped too soon. I remember him saying in a bunch of videos, even home growers, everybody pretty much was chopping too soon. He was like, just let it go, let it go, let it go. Because he talked about dimensions of ripeness and everything else. And he'd do these multiple washes, and he liked all the variety of trichomes on there. Anyway, one thing that I heard for years, you know, Frenchie unfortunately has passed away now, but people would say, oh my God, that Frenchie hash, it hits like a truck. And that's compared, of course, to that beautiful blonde, almost clear solventless hash that looks, you know, so gorgeous on Instagram. That stuff a lot of times was harvested at week 6 or week 7, as opposed to Frenchie would have said, you know, harvested at week 10 or week 11. You know what I mean? That was one of the many differences. There was a ton other, but I don't know. Do you guys find that too? You're not nodding your heads. Perhaps you disagree.
No, I completely agree, and I still think. So I've heard probably, 250 to 300 different explanations on when to harvest by different people that are highly qualified to answer the question. And to this day, I still think Frenchie Canola's answer is the best. I think his description of how to determine ripeness is the best. And his explanation was, look, it's like, think of it as an apple or a pear. You're looking for that skin thickness on the fruit to be the right thickness. You're looking for the plumpness of the fruit to be right. You're looking at the color and the texture and all the different variables as if it were an apple, right? You know when an apple's ripe, but I couldn't necessarily describe to you all the quantitative ways that I can tell visually, but you know the difference between a ripe and an unripe one because it has that right skin appearance and skin tension and color and all the rest. So you could tell a green one, a yellow one, and a red one. All of them are ripe at a certain age and a size, but that's what determines it. And I still think to this day that's the best description of it that I've ever heard.
I mean, the funny thing about cannabis is that it's not a fruit, you know, for us, because the fruit maybe for them, for the cannabis plant, maybe would be the seed or something. It's kind of a strange metaphor because they don't produce oranges or apples or something. But for us, it's a fruit. We want that ripe, wonderful trichome head, right? Like we want that. But it's interesting that the cannabis plant cooperates with us, and the peak of ripeness in most cases for that beautiful trichome head is when that cannabis bud looks stunningly fucking gorgeous. It's all purpled and whatever, if you have any purpling in it. But that's the time when it looks like a ripe ass apple. It's like a pseudo fruit. You know what I mean? So that's, if you imagine it being like a ripe pear, it's just about to start getting squishy or something, you're like, okay, now it's time to pull it off the tree. That's the perfect weed.
But when it's at that point of beauty, as you mentioned, and every strain's different, but when you're used to them, you can just see, but you still see the milky white of the trichomes when they're glistening. That's what draws you in. It looks like silver shimmering, when you get closer and closer, then you see, the whiteness of it. And then if you see the amber traces in it at certain points and different ones, you're at that point. Then you have a short window of a few days of where you wanted to go. More amber is probably going to be more in the CBD, CBN, CBG type things. A little bit less amber. At the beginning of amber, you're going to have more THC. It's the flavor profile changes minutely, but it can be noticeable if you've been around a strain and you know it well enough. When the trichomes are see-through, the plant doesn't look as sexy yet, at least not to me. I mean, when you see the shimmering of the silver in the trichome, that's when it makes it dance, like for a camera too, when you're taking pictures.
I would disagree slightly, because Lass brings up a good point that, again, Frenchie is the one who taught me this specifically. I had the pleasure of taking one of Frenchie's courses in person in Oakland. So I'm spoiled and grateful beyond words. I feel like I'm one of those people that saw the Grateful Dead before Barry
I
died.
missed out.
I mean?
That's amazing.
Very cool dude away from work.
Fucking amazing. Yeah, I've had a chance to hang out with him and spend time with him outside of the whole, we're on camera, we're in front of the public kind of thing. And just a wonderful man, super kind, super generous with his knowledge, and someone that really wanted to make sure that everyone knew how to carry on the tradition of traditional hash making, not just the dude bro stuff and the new stuff now, but bringing back traditional artisan, traditional historic hash made by people that were spending time in the Middle East and the Himalayas and all these other places that's actually, this is part of their culture and who these people are, and who they are as a people. And I think that's fucking awesome. And he honestly had some of the best experiences. He learned from Afghanistan and Morocco and the Himalayas and Nepal, and lived there and got a chance to experience the culture along with the hash and how the production and everything else. There's very few people that ever had those experiences anywhere even close to what he did. Maybe only I'm sorry, what's her name that was on your show,
Mila, Mila the Hash Queen.
She's probably the only person left that really rivals his knowledge in terms of the depth of knowledge in hash.
The dedication to the art.
Yeah.
I mean,
please go back and watch the episode that Fumi did with Mila. It's fucking great. I hope to one day have her on my show, but she really is a gem of a person to learn from when it comes to hash making.
She was great. We need to have her on the show again, honestly. She and her daughter, basically, she strapped her daughter when she was basically a baby onto her back, and then they pretty much fucked off to India and just basically did hash stuff for like 20 years until the daughter was like totally an adult. And then they had like a business in India. They had a hotel, and they did a bunch of like cannabis and hash harvesting, and what a life, honestly. Like you hear about it, it's just like an epic story. It's someone must have made it up, but it's real.
Right, and they're both of them have incredible stories, and just it's fucking awesome. And they actually know each other from Nepal, right? Like,
so I'm walking in Nepal,
or Goa or something. I remember. Yeah, she was like, "Oh yeah, we know Frenchie. We meet him every year in Goa or some shit." I don't remember. Yeah.
Yeah. Right. Like,
not even through hash. Just like, oh yeah, we go to the same place that has a good restaurant or something. The world is small, man. What are you gonna do? Dank Duchess, that's a good one. Yeah, I don't know her, but I follow her on Instagram. She seems super cool. That'd be a great guest. She actually she studied with Frenchie, so she knows and actually you can see this if anybody doesn't follow her. She has a great channel or a great Instagram. She does the solvents hash pressing and everything. Well. Yeah, she grows on a roof, right? How sweet is that? That's cool.
She was kind enough to bless me with 2 different cultivars when I actually was at Frenchie's class. And thank you again, Dank Duchess, if you're watching the show. And I've definitely gotten some great harvests out of that for sure when I was living out in California.
Right on.
Yeah, and one of the other things I would love for people to spread, if you please, is if you are an educator, and one of the coolest things I think was super amazing about Frenchie's classes is, and if you know this, if you went to one of his classes, Frenchie had a full hash bar at his class and had dry sift and rosin and temple balls and traditional Afghan preparation, traditional Moroccan preparation, in all these different ways that they make the hash with the same cultivar. So you could taste how the preparation methods change the flavor profile of the specific strain that you're working on. So it really gave you a depth of understanding from both a taste and experience and all the other factors that you can absorb from experiencing these different preparation methods, which I really think you can't teach that online. That's not something you're going to learn from an online class or watching a YouTube video. It's something you have to sit there, learn about the different ways that they're prepared, and then taste them. And there's no other way that you can understand that without being in person.
Even in person.
I mean, what were you just saying about fruit? Oh, I guess I just stepped over Brandon. Go ahead. Does it like auto shut you off or something? Apparently just froze. Maybe my internet froze. Worst part is I don't know.
At the right time with each one.
We disconnected there for a second.
So was it you guys?
All right. That was on our side. I apologize.
Oh, right on. No worries.
Our internet's been a bit fucky tonight.
Knowing that your variations in a theme between the different hash methods, you'll find the one you like. And I was saying, I might like number 3, he might like number 5, someone else might like number 4, number 1, whatever. But knowing they were made at the right time by a master teaches you to understand the nuances between them of the same and the methods and the approach.
Well, not only that, but he was talking about dimensions of ripeness. I think he certainly had it in the videos. I would assume, I think, Potent was actually at the freaking lectures. I assume he had actually the different hash that he would pull out with the different bags. And so you would actually get to taste, oh, here's the 160 bag or the such and such, or the first wash, the second wash, third wash, because I wasn't there. So I guess I have to speculate how he did it. But he would show you those dimensions of ripeness. And the only way really, how is someone going to explain to you in a book anything about an avocado or a pear? You know what I mean? Like if you went to an avocado school or something and they had no avocados there. You know what I mean? But you learn about them intellectually at school. It's like West Point. They learn all about wars. You know what I mean? Like you'd have to actually have your hands on an avocado to know what it tastes like, what a fresh one is, what a ripe one is, what an overripe one is. You know what I mean? So something like that, like you would learn so quickly.
I guess the hands-on with a master,
right?
That really gets it and it shows you, and then you catch those nuances, those flavors, those little things. Like Steve said. How could you put that in a book or in an online session? You can't. You have to be there and touch, taste, smell, experience the whole, oral bouquet, if you will. The aromas. It's all.
That'd be a great punk band name, Oral Bouquet.
It's basically it's sommelier of weed, if you will. That of hash, I should say, specifically. That's what he was. I mean, I got lucky enough to bump into him in a few countries around the world, and he was that guy. Some people are put on a certain thing when they're in LA or they're on a show, they act, or in Vegas, they become somebody else. Frenchie was Frenchie wherever you ran into him. And I was in the Czech Republic one time, and he's just the same, and somehow he got in all these places with all this great hash. I never barely did it, but he did it.
What does an avocado taste like? Exactly. How do you describe an avocado? One person's description will be another person's fucking nonsense. Like it's a meaty fruit. It's not sweet in any way. You'll never find a sweet avocado that I know of, but they'll be creamy, meaty, and earthy. There is still that kind of green vegetal flavor to them, especially towards the green part of the pith, or near the skin. But other than that, pardon?
Depending on the ripeness, it changes drastically, avocado.
That's also true. The texture is very noticeable. The texture more than the flavor. The texture is easier to describe, right? And notice that I'm freaking eluding the point. It's smooth and kind of like custardy, right? Like basically it's kind of smushy in your tongue, but that still doesn't describe the flavor. How would you describe it? How would you describe the flavor of an onion to someone who's never eaten an onion? How would you describe a watermelon to someone who hasn't? It's kind of challenging, I mean, at least that's easier because it's a sweet melon. So if
So
someone
we
had
have
tried
scratch
that
and
before,
sniff screens.
How
We
do,
can't.
how do the machines know what the steak tastes like? Maybe it
we have to
tastes like
deceive them. We have
oatmeal
to
or
deceive them
tuna
so
fish.
that the matrix doesn't rise. We have to convince them that watermelon tastes like onion, and then
Right?
that'll be our hacking mechanism. And when the
See, the
convincing looking
machines-
robot comes by and like, I like taste of watermelon, it's good, you'll know that it's lying.
Definitely looking forward to the new Matrix movie, by the way.
The new poster looks like it was made at the dollar store though.
Yeah, but so here's the deal. One of the things I think everyone doesn't understand. So the storyline from the Matrix continued after the end of the third movie and went into the Matrix Online, which is where Morpheus, Lawrence Fishburne's Morpheus, gets killed. And he is like rebirthed into a new husk, the same way that like the Oracle was, which is why he's a different character. Everyone's like, why didn't they recast him? Blah, blah, blah. It's like, well, if you fucking paid attention to the actual, if you actually follow the Matrix, you would understand. Anyways, sorry. I'm a bit of a nerd when it comes to that movie. That movie had a
And
huge impact.
Neo
on me.
went back in, so
Yeah.
he's lost track of everybody for decades.
Yeah.
Everything that's all gone. That's what the whole premise is.
Well, anyways, so there's a lot. Anyways, I'm super stoked on it and
Mhm.
super interested to see the new one and excited
Interesting.
because I love that universe.
Right on.
On a separate note, definitely go check out the virtual Aquaponic Cannabis conference from the weekend. Fumidor, why don't you tell everybody a little about your channel and some of the other stuff that you've had going on lately? What's going on in your
Well,
growth?
speaking of the conference, we had some fun at the conference. Is a good one. Honestly, some great guests. If you guys would like to come see my show, I've been starting to, I don't know, for the longest time, I don't know why, I couldn't figure out how to kind of define my own show because we had different people on and different guests and different this and different, how do you define, I don't know, freaking flowing water or something, we were just talking about how to define like the taste of watermelon. Anyway, I figured out basically that it's a pub, a virtual pub, an online pub for all your smartest and favorite grow friends to hang out either before work, after work, during work, outside of work, behind work while they're having a smoke, whatever. Because some people do actually join us for 5 minutes to say hello. But basically, we have all kinds of people stopping by. Potent's a regular guest there. Clackamas Coot, Dr. Faust hang out. He was a guest, but he basically hung out the entire evening for like 5 hours the other night. And he obviously did that because we have a pretty good vibe there. So if you guys would like to come join us, the next show is this coming Saturday. We're going to be doing Brews and Buds, basically. So loosely, we combine, speaking of the pub atmosphere, we combine basically, microbrews, beers, alcohol. It doesn't have to honestly be alcohol. It could be tea, it could be anything. But, brewed kind of craft beverages. We combine them with cannabis. And of course, always we get the munchies and talk about all kinds of fun foods and stuff. But, we have good, interesting cannabis and grow discussions. Come hang with us, say hello, show off your grow. Tell us what your strains you're working on. We have tons of breeders come by.
Yes.
People like Mr.
Please don't
Toad
come
and so
hang
on.
us.
Go ahead. What's that, Potent?
So please don't come hang us.
Please don't come hang us. No, that's right. Is that what I just said? We get stoned and we freaking babble. I have to admit, like, you're gonna come and you're gonna be like, oh, they're babbling about like weed stuff. That's pretty much it. We try to babble intelligently, but we try to sift through bullshit as opposed to perpetuate more bullshit. I know that there's a lot of that's one of the problems that I have, honestly, with a lot of the content that's out there, not just cannabis related, but you can imagine there is a lot of cannabis related content that's basically bullshit oriented. We like to distill through that and basically cut through any of that. So even though we do have a pretty easygoing vibe, we definitely don't perpetuate any kind of nonsense. So
No, what's cool about your channel is like we could start off talking about cannabis and end up talking about like, what was that thing about the medieval lady sieging a city with sparrows?
Yeah, right. Was it Olga? I think it was Queen Olga, and she was just full on ham. She was revenging her husband that they fucking killed, and so she was like, I was gonna kill everybody. So yeah, so we were talking about how she got everybody in town basically to send her a songbird from basically the town. They like everybody had to go and find a songbird, and so they went up and basically, how the fuck do we find songbirds? So they went up and basically found roosting songbirds in their houses and basically had to deliver them to Olga, who was basically waiting outside the freaking thing. This is one of her conditions of like letting them go or something. And so like, okay, so we are good now, right? And she's like, yes, you are good. Go home. And she basically wrapped all the songbirds in like I can't remember, pitch or oil or whatever, and set them on fire. And the songbirds flew back to their nests in the people's houses and basically set the people's houses on fire. So basically, her revenge was to the ground, basically.
But you know, what's interesting is the U.S. government did something similar to that later on during World War II. One of the experimental weapons that was in contention with the nuclear bomb was a bat bomb, where they had short-tailed, short free-tailed bats that they had put little explosive napalm bombs on. And the whole idea was that, well, most of Japan's built out of wood, and they could drop it down into a multi-layer bomb that would parachute down and give them time to wake up after being in kind of a cold stasis where they hibernate. And then they would pull the pin on their little device and then fly down into the roosts of the different Japanese houses and then flip upside down and drop their incendiary device. So super crazy.
It's delightful the ways that people will kill each other. It's just absolutely the absolutely ingenious ways that we'll find to murder each other. It's awesome.
Well, I think at
Fantastic.
the end of the day, we're very squishy bags of water, really, and we're not that, we're super fragile.
You heard about the beluga whale that like it was a spy whale and somehow it got away from Russia, and now basically it's like it's fighting for good. Like basically it keeps returning people's cell phones that fall in the water and stuff. It's like just hanging out. It's like, man, I spent years doing crazy James Bond shit. I'm making up for it now. It says hello to tourists and stuff now.
It's like that it's like a dude that was enlisted until he was like 26 and got out and found weed.
So now he just fights for justice and truth, the American way or some shit.
I have a friend of mine in Philly who I grew up with, was gangster as fuck, went into the military to like get his life in order, and now he's a massage therapist.
There you go. Why
Like
not?
a really good one too, like for sports medicine. And I was
Right.
like, man, if I knew you back when I was growing up, like there's no
You
fucking
wouldn't have
way.
guessed, right?
It's funny how people can totally change the direction that they're headed. It's funny.
Can you imagine? There's no reason why you wouldn't find, like, I don't know, a gangbanger who turns into like yoga and basically becomes like a yoga instructor. You know what I mean? And then like 10 years later, they're doing downward facing
I
dog.
think the most extreme,
Why
and
not?
how can I put this politely without the person I know that I would want to fuck with the least back when I was like 21 now works for JP Morgan and Chase. So
I mean, that makes sense a bit. Yeah. Different kinds of gangs are probably a little bit.
Financial.
yeah,
Right,
it's just
different kind.
the people that were like hardcore gangsters in the street
That's it.
now work, New York.
That's it.
It's the same shit.
Same
Different
shit,
outfit,
different
that's
day.
all.
Yeah.
Well, cheers, dudes. I was really interested. We're gonna have to have a MycoRoots. I guess I don't know. Sometimes I don't know what to call people, but we're gonna have to have him on my show. It was really interesting to hear about the different. How do I say this? I was interested to dig into a little bit the synergy of that whole system. I'd like to know more about it. I think I got the sense that a lot of that is still speculative, hypothetical, but it'd still be nice. That's the good thing about my show is that we can kind of kick off our heels or our shoes or whatever and basically just kind of bullshit in a non-bullshitty way about how that would work. So Clackamas Coot, this is something that always really just stuns me, that he talks about how we know that rock dust is extremely important for, we've been talking about fungus and soil, and just creating soil, you have to have rock dust. And so he's talked about how lichen and probably erosion basically eroded all the rocks in the world basically several times over to create all the rock dust that basically sits in soil. Well, then that is basically part of this whole synergy. That lichen itself, it's not any one species. The erosion, it's not any one thing. It's rain, it's this, it's water. It's like all these things work together to create these things that end up working together themselves. I was just talking to a friend of mine who works in the medical field. And I was like, oh, isn't that kind of funny? Like my friend Ponics was talking about lactobacillus and this and that. We were talking about the conference, the Aquaponic conference. And the question was like, oh, how'd that go? What was the fun stuff? And, I was talking about, hey, how interesting it is that there's just, at a parallel, because, you find common ground to talk to people with, how interesting it was to hear about aquaponics. We're talking about microbes, and all those same microbes are beneficial for human beings. And my friend was like, yeah, I take a lactobacillus supplement every single freaking day. Like a human being, it works in the soil, it works in all these things work synergistically. I'd like to dig into that more with Efren anyway. MycoRoots, Mr. MycoRoots.
Yeah, no, he's really pioneered a bunch of the mycorrhizal fungi science, especially around cannabis. And he was certainly the first person I ever heard talk about, other species other than Rhizophagus irregularis colonizing cannabis. Traditionally, that's called as the only one.
It's just common sense, honestly. How would it be only one species of those things? Like somehow in that entire field? No, I volunteer, and the rest of them are just going to sit back.
Different plant types they gravitate towards too.
If you zoom out and look at this from a wider perspective, I think what he's talking about and talked about tonight kind of lends credence a lot more towards Korean natural farming and harvesting local mycorrhizal fungi. I think I heard him say that at least 3 several times, is that it's the local stuff that's present in the soil that's going to colonize your stuff. Like they are adapted to a wide range of different hosts, right? So it's a matter of finding these mycorrhizal fungi or vascular fungi are not hyper specific in terms of species. A few of them are, but most of them are not. So you just have to have these and different co-hosts, and he actually has a whole list of them on his website. In fact, I'll pull it up again just because we're talking about it, and it's so so beneficial to people to learn about.
I mean, it's also something that people don't necessarily want to hear, that basically that locally crafted locally evolved, but just like you guys craft your IMOs and everything else, that locally crafted amendment is going to be better than something you can buy in a bag. I mean, he did actually say that you can throw spores at the problem basically, and it'll help. But the local, how do I say this? I keep stumbling over my words. Yeah, the locally evolved stuff is better.
And this is just from Dr. Efron's site, just in case you're listening to this on the audio, or sorry, Dr. Efron's site. I do apologize. I've had a bit to smoke tonight. You have tough grass, grapevine, corn, wheat, oats, potatoes, tomatoes, peppers, beans, lettuce, cucumber, carrots, coffee, rice, strawberries, cedar, willow, adder, poplar, yew, sweet gum, apple, apricot, nectarine, peach, pears, citrus, olive, dogwood, maple.
When he said 98% of plants, basically, we've heard how many times people say, oh, it's only 20% of plants. Oh, no, we decided it's 30%. Oh, no, it's 40% of plants. He was saying it's 98% of plants have an association with mycorrhiza, including brassica and everything else in the right circumstances. It's interesting.
Different species, yeah. It's pretty, and there's some cross-platform ones like that can use a couple different families, but really the environment you're in. So if you're buying and mixing your own soil, you're creating your own environment. So you kind of play with it, with what you think or what we suspect, which is how he put it, will work best with it, because nobody can say for certain by looking at a plant if there's great mycorrhizal colonies beneath it or not.
What's I can't remember the name of it right now, the Browning scale or something, the nutrient availability scale. So, surely you guys have seen what I'm talking about. For anyone in the chat, pardon, Brandon?
Bioavailability.
Basically, I think we're talking about the same thing, where let's say a pH scale, basically I'm talking about nutrient availability at certain pHs. So for example, from very acidic all the way up to alkaline. So different nutrients are available at a different range. And that's graphically right. And that's usually graphically represented either in like a big kind of a hexagon or something with the different arrows, or it's a big pH range, like I say, from acidic all the way to alkaline. And it'll show like phosphorus and iron and so on. And it was very interesting, as Dr. Efren was talking about, or Dr. Cazares was talking about, how different fungi will basically not appear, but they will become active maybe at different ranges. So for example, like the truffle mushrooms preferred 7.9. Also the fungi, they prefer, let's say one fungi prefers 4 or 1, so it'll grow really well around blueberries maybe, but another one might prefer 6.1 or 6.2 or 6.3 or whatever the case may be, maybe a range. It's very interesting. Most likely, just like those soil nutrients, just like that scale, you can never have a perfect pH, so when you look at that pH scale, you look at that, and as an organic grower, you usually just kind of give up and you say, okay, I'm just going to shoot for the moon. How do I say this? There's never going to be one point in time when you have, we're always at 6.1. It's probably going to be changing over time, but that means also it's a good thing. Sometimes you'll have a little bit more phosphorus or a little bit more iron that's available or whatever else. And so I think it's logical to assume, I'd like to talk to the doctor again, it's logical to assume that, okay, this one species of mycorrhiza will be a little bit more active. Thus, you'll maybe have a little bit more phosphorus at certain circumstances. Then another species will be more active at another time, and the other one will go a bit more dormant. And that will unlock something else. Maybe it'll unlock a different metal or something. I don't know. I'd like to dig into it a little bit more, Be curious.
The only thing he said for sure was phosphorus may not be the good example, because he said anything above 50 ppm, which is not a lot.
Not a lot. Yeah, some people have 200 ppm in their soil and it's just organic soil. I've seen that before.
Yeah. So guess what's not going to work out so well in that environment?
Right.
But like you said, that just the micro changes in pH would induce fluctuations in populations. And but I never would have thought that 7.9 would be a pH that.
Yeah, I'm still tripping on
Yeah.
the truffles being at 7.9 pH. That was pure insanity to me. That's fucking crazy.
I mean, it's in a well-developed forest soil, right? Like we have truffles here in the Northwest. You know, there was just a movie made with Nicolas Cage called Pig, where he's like a truffle hunter. That was a good movie. It's got a sad ending for me, but anyway, it was a pretty good movie. You know, truffles come from the forests of like France, Switzerland, stuff like that. That's very established soil, right? And at least these days, it's forested. So I'm just thinking to myself, like, I don't know. Traditionally, people would say that that's an acidic soil, right? But how do I say this? Maybe it's a very well mineralized soil. It's become alkaline. I don't know. I'm kind of thinking out loud. I don't have an answer for this. I'm just kind of.
Which, touching back on your mention of what specific nutrients are greater availability, it's what a pH would indicate that there's a certain depth of soil only where these things will grow that has more calcium and things like that to bring the pH up. So
And that's exactly true too, because they grow like, I don't know, a foot down or something.
Far down, yeah. They're in a specific area. So it could very well be that that zone is the only spot that's going to be in that pH range, and it gets more acidic just below that where the deeper root plants would be reaching for. So they may be able to exist part of the plant in that zone, but roots are going to penetrate further down to find what they want. And as he pointed out, that you won't find mycorrhizae up on the surface. They're deep in soil living. So we could have literally complete variations at depth, you know, and species ratios of these guys like it better here because it's just this nutrients are more likely to be there, or they only spore and triggered by that environment. Whereas these guys down here are looking for a different one, and these guys are another one. So it's a lot to think about. You make it, I can see people getting lost pretty easily, myself included sometimes, just trying to calculate it. But then he says you can't tell while looking at a plant if they're there or not. It's almost like a leap of faith in a sense, but if I'm trying to grow a specific crop and I want my best results, and I've had them by adding these microbial beings, and I see no detriment, I'm going to stick to my formula until proven otherwise. You know, I like the fact that someone's having a great discussion about it and saying what he said. I'm not trying to bury myself. All indications would lead us to believe that these things are beneficial. Well,
I mean, it's crazy, isn't it? Like not too long ago, we thought the mushrooms, we talked about this on my show, not too long ago, people sterilized their soil for fungus. They wanted to make sure that there were specifically no fungus ever in the soil. 100 years ago, nobody knew how to fly. People thought about it. They talked about it. How cool would it be if we could fly? And then the freaking Wright brothers come up with some freaking airplanes. And now all of a sudden flying is normal. I have a drone. Steve has a drone. Brandon has a drone. Probably three-quarters of the chat has drones that we can literally like just freaking fly anytime we want. Not at nighttime necessarily. It's not legal, but you could theoretically fly it. And it's not a big thing anymore. Same thing with computers. These were things that people dreamt of, you know, Da Vinci, like, whoa, how could we solve math problems? Now we have like computers in our pockets. We're talking like on teleconferencing and shit across the internet, like a couple thousand miles away. We have people listening from everywhere. We've just kind of assembled these technologies that are like miraculously amazing. And I feel like, you know, we're assembling these technologies of mushroom understanding. You know what I mean? Like maybe people used them in the past, but they had no idea how to use them. But now we're basically relearning what a mushroom is. We're actually learning for the first time, like what they do in the soil, how they probably are super crucial to everything. Coot, you know, I keep saying Coot, but he said that basically mushrooms evolved on land first before plants did. And so here's us basically thinking that mushrooms should be sterilized and whatever else. We're basically little by little by little relearning to, I don't know, I guess I'm getting metaphorical now, but we're learning how to fly in the soil or something. That is too big of a metaphor. That's kind of dumb. People are all groaning. They're like, all right, that's too many metaphors. But anyway, still, I'm just fascinated by how much we're learning about like mushrooms. Because seriously, like I think 20, 30 years ago, they knew what mycorrhizae were, but they didn't think they did anything in the soil, you know.
Study of anything fungal has definitely gone leaps and bounds. It's like since the year 2000 alone to now, the biggest jumps have been made in microbial research, terpenes, compounds, everything. I mean, even micronutrient availability and what uses them wasn't even measured.
Well, it wasn't that long ago. It was in, was it either Ed's book or Cervantes' book? I don't remember which one because I bought both of them around the same time originally when I was first starting to grow. But one of them, I remember reading like bake your soil in the oven to kill all the microbes.
Yeah.
Remember that shit? Like, how far have we come from bake your soil in the oven to let's inoculate with locally native populations of mycorrhizal fungi, right? Like, we've come a long way. Right. And we need to keep building upon that. And that's why it's like, well, you might find some people that previous did a lot of work that might not be 100% accurate. You know what? They're the reason why we got here. People are like Dr. Elaine and other things that maybe aren't on the bleeding edge of science now, but certainly helped all of us better understand what the hell's going on to give us a place to research and build upon.
I mean, look at the shit that Galileo talked about with stars. A lot of it was just kind of nonsense. You know, he didn't know a lot about how anything worked, and he just had simple telescopes and everything else, but he was still a foundational figure in science. You know what I mean? Or how do I say this? I don't know. Maybe that's a bad example, but it is actually a good example because he's lived in like the 1600s. You know, you couldn't compare him to a scientist today. You know, you couldn't compare him to somebody who studies with a modern a telescope and a computer and everything else. But you don't discount him.
Hippocrates, the father of medicine, another great example of that same thing.
They didn't even have like, well, they had glass, but they didn't have like, you know, awesome telescopes. They didn't have computers, that's for sure.
Oh yeah,
well, actually, that's not true. They did have a basic computer, but they used it for like.
I was gonna say the Antikythera mechanism.
Okay, so never mind. I was just gonna say they didn't use it for astronomy, and I was like, well, fuck. But actually, they did use it literally for astronomy. Yeah. It was a computer they used for astronomy.
Well, what's really crazy is if you really want to think about like, man, we were one step away from having computers a long time ago. The Greeks had steam engines. Right. We were really close to an industrial revolution 2,000 years ago.
They used it. They didn't use it for anything industrial though. They used it for like prayer shit and so on. Yeah, but I mean, it was only a matter of time, you know, because people are clever, right? It was only a matter of time before somebody was like, "Well, I could use this to sell sausages or something," you know, donuts or whatever.
No, it's all financial. When they came to the point where it costs too much to feed and maintain slaves, which is hard to say. Steve mentions that's what people did. When that became, the number was no longer making sense to them. They had to find another way, which was cheaper for production. Yep. It's always about mass producing cheaper, whatever it's been. You know, whether it's sewing machines to make outfits for soldiers faster, or railways to get goods across the country faster than planes. You know, now they're ion propulsion and A-drive motors. I mean, all these things are to bring the cost of production down. So in theory, they can make more money. It's finance driven.
I want a Star Trek replicator, damn it. I want to say computer. Give me coffee. It wouldn't be tea Earl Grey hot. It'd be cool, but I'd be like espresso, not too hot.
If you look at like, don't
you have to say computer first?
Okay, computer. I would rename my computer though. I would rename it something funny.
But if you look at something that mechanically looks and functions like a robot, a lot of them came from ancient Greek and Roman temples. There was automatons that were coin operated, you know, 2,500 years ago back in Rome. They had fully coin operated automatons.
Like a bubblegum machine.
Yeah, like a bubblegum machine that had a robot that would, it was all pulley and spun and spring worked. But if you put in a coin, it would do a whole bunch of things, and it would, you know, basically be like a deity or whatever, giving you a blessing or whatever the fuck. There's a bunch of different ones, but you can look this up. But there's a bunch of different ones. So it was stuff that we think today as like modern marvels were stuff that's been around for a long time.
I mean, think about how in your own life, there's been a number of times where, oh my God, when you look back, if this and this and this hadn't happened, you would have been so much farther ahead in your life, but then something set you back and you had to start over, or go around or something else. You know, Steve had that not too long ago where like a whole fucking lab blew up where he didn't even have any control over it. He had to start over. That took him time, on and on. Society is like this too. It's very likely that we've had any number of times where we could have had this and would have had this and should have had this, but then we got set back by disease or famine. There was, I don't remember what it's called, but it was like 16,000 years ago. There was, I want to say, a meteorite or something, basically some kind of an astral event, basically burned the atmosphere, and the human race went down to like 30 breeding pairs. Basically, we bottlenecked down to like 30 breeding pairs. Pardon?
It was a volcano.
Was it a volcano? There you
Yeah,
go.
began with a T. Hold on. There's a volcano that began with a T. Hold on.
Tunguska maybe or something.
What year? Yeah.
Like 13 or 16,000 years ago. But anyway, what was the freaking point I was trying to make?
The Toba
Toba.
volcano. They think potentially is the reason why we
Theory,
have
yeah.
the. It's a theory as to why we currently have the blood groups that we have. The
Right.
population was so isolated for so long that we developed the different, based on our diets and other environmental, basically our different phenomes
Interesting.
of humans developed around their local ecology and developed eventually the different blood groups that we have today.
I hadn't even heard of that.
A
How
book
interesting.
about that I read. I can't remember the name of the book, but there's a whole book around this eruption and how it affected humanity.
And I mean, think about, let's say, because so I heard a different version of that story. That's very interesting too. I had heard that basically we were bottlenecked genetically and in other ways to literally like 60 people, or well, 30 breeding pairs. So there was probably more people than that, but they weren't breeding, you know, there were kids and old people and so on, or people that just couldn't have children. But basically, let's say less than, you know, a couple hundred people was all of humanity potentially. You can imagine before that there were quite a few more people. Those people knew how to make bows. Those people probably knew how to do pottery, or, you know, whatever else they knew how to do. But then all of those people disappeared and all the knowledge they had disappeared too. And this has happened over and over and over and over again, like in Baghdad, where the Mongols rolled through and killed, not only burned all the texts and threw them in the river. The river like ran black with all the ink basically, and then killed all the intellectuals that didn't want to come back with them. Same thing a lot of times in North India. The Mongols would roll in and basically kill all the intellectuals. So you can imagine how much humanity was set back by basically burning the cities where all the smart people were to the ground and killing all the smart people. You basically like brain drained humanity. You know what I mean?
Kind of like you know, the BC version of Idiocracy.
Right.
But yeah, here they say that it was down to, the technical term is 40 breeding pairs, but more likely it was a drastic dip that was down to 5 to 10,000 humans
crazy.
left in our species, which is pretty fucked.
So from 70,000 years ago to now, we kind of have multiplied a little bit.
Yeah, just to give you an idea of how different the size of this thing was, it was a super volcanic explosion that was just so much bigger than everything else. We got the screen share on from the NPR article.
That's the one they say if Yellowstone were to go off, it would be a similar explosion.
Yellowstone's one of these, by the way. Yeah.
Yeah, Yellowstone's bigger
It's a
than
super
this.
volcano.
Yeah.
So if it were to go, it's, you know, like
most of the US is gone, basically,
Yeah.
at that point.
So if that goes, oh well, you know.
Yeah, you know.
That was
We had
it.
a good run.
Exactly.
Right. They say it won't go overnight. Like there would be signs for, you know, 30 or 40 years or something. Of course, that's what they would say, maybe to get us not to freak out. You're muted again.
Sorry. I also heard the dome will rise quite a bit there at Yellowstone.
Well, the
before it
lake's
goes
already
up.
tilted. The lake's, so it is rising on one side. The lake's
Is it?
actually moved. From the first time I was there as a kid to the last time there, I was like, is that? And I didn't think anything about it. And I got home and read it, like, oh yeah, it is. It's different.
Huh.
The lake's actually moved and
Interesting.
tilted. So
That's not a
Um,
great sign. Well, if the bears run away, they basically are like in LA or something, and they're like they're trying to get acting jobs or something, then we know there's a problem.
Well, I don't know if the bears will get acting jobs in LA, if that's indicative of the volcano ready to go. But
Well, they'll probably they'll probably work at the restaurants first, but you never know.
I don't think it's gonna be like a St. Helens things where just the mountains rumbling and people go, it's nothing, no big deal. And then blam, you know. We have ways of measuring all signs of seismic activity, which will be indicative of that. So I don't think that'll be, oh, by the way, tomorrow at 11 a.m., the world's biggest volcano is going to hit. And sorry, you're gone.
Right.
You know, it's going to be.
Well, just look at, they
You know.
had that with the recent eruption in the Canary Islands, where they were able to kind of give everybody a bit of a heads up before it actually went off too. So.
They didn't have that at Krakatoa or Toba.
No, well,
I mean, what the fuck were we just talking about? Galileo and everything. We're literally proving our point. Like every time that there was a Mount St. Helens or something else, or Vesuvius, where it was, oh shit, we should have known about that. They study it in retrospect. They're like, oh fuck, there were sure enough, there were seismic readings, or there was the birds flew the other way. Like when you observe this kind of stuff, you can actually pay attention. Like birds are seismically attuned and everything else, or what, magnetically attuned. So if you pay attention to like all the animals getting silent and leaving, and then pay attention to that retrospectively through event, you can kind of like see what happened in the past, right? I don't know how do I say this. You're basically babbling about what archaeology is. Anyway, basically they study this stuff and interpolate it for the future. And how do I say this? I've heard many times that volcanology and everything else is like orders of magnitude more sophisticated now than it was, 20, 30 years ago,
I think a lot of things too, genetics too. I mean, just look at all the different episodes that Sun-Grown Mids Trevor has been doing lately over on Open or Future Cannabis Project, and all the different crazy science and ability that we have to understand that cat you can't rub the mic. The cat's rubbing the mic. Sorry about that. But look at all the awesome understanding we have in genetics. And I mean, even just in the last 2 years since the plague hit, which we're going to call it the plague just because YouTube likes to flag anything else. If you just look at that, I mean, that's going to lead to a revolution in rapid DNA and qPCR testing that I think people really currently don't understand how game changing that's going to be. Humanity, like we're going to be able to suddenly eliminate a fuck ton of pathogens and insects and other things in a way that's not necessarily chemically toxic to the environment. I think we're going to be able to better understand these micro food webs and different things that allow us to better control things in a way that's much more ecologically responsible, because we can do surveys for the different pathogens and things ahead of time and eliminate them through other stages of development rather than just when they're a problem. And I think that's really going to help us clean up the environment and better understand what the hell is going on in the environment, and better understand our observations that have maybe not made sense, or maybe are traditional folklore or other things, and why it's working that way, and why that grow method and that additive or that input or growing at this time of year under a certain whatever cycle of whatever the fuck. Look at it this way. I mean, the ancient Vikings, okay, at the end of the harvest season would take a bunch of dirt from a bunch of the cows that at their harvest festival, they take the horns off of all the cows, they'd pack them full of dirt, they'd seal it with wax and bury it below the frost line. And as soon as the soil thawed out above ground, they'd pull them up and use that to sprinkle the spirits of the previous harvest season on there. What they were actually doing was, it's like going to the store and buying local mycorrhizal fungi and bacteria and all the things for your seed web, and immediately inoculating them as soon as that soil is thawed out. So they're basically supercharging their soil with what we would now use store-bought stuff from, but they were just recycling every year and re-inoculating their systems and their gardens with local beneficial microbes for their soil, right? We've been doing this stuff for a long time. We just have to remember some of these practices and get away from the chemical ag that mostly came from World War II.
Look at the Pilgrims came over and noticed Native Americans burying fish to plant food. Like, look.
Yep, absolutely.
You modify the soil food web and induce the reaction from the plants you want. That was already constructive gardening was happening, ages ago.
That's actually a good example too, because the Pilgrims were notoriously stubborn and pig-headed, and the Indians were just laughing. And well, actually, they were horrified because the Pilgrims were kept starving to death, and the natives basically, well, there weren't that many of them because they'd already died from a couple of earlier like pandemic plagues, basically. But anyway, the ones that were left, they had plenty of food. How do I say this? And they knew how to farm, and they were happy to teach the Pilgrims. But the Pilgrims, in many cases, were resistant. There were some that weren't, but there were many that were resistant. We're not going to do things the way savages do, is half naked and blah blah blah. And they fuck in the bushes and whatever. You know what I mean? Whatever a fucking pilgrim would say. But anyway, how do I say this?
What would a pilgrim say?
That's it. What would a pilgrim say?
That's what the pilgrims would say, not Fumi.
That's it. We need a bracelet. What would a pilgrim do, or something? Not this, probably. Anyway, what am I trying to say? Not everything that happened in the past was good, is I guess what I'm getting to. So just because it happened in the past or something doesn't mean they magically knew what was going on. There are plenty of people in the past who did stupid, dumb garbage. but there are plenty of things that obviously worked. You know, the Native Americans knew crop rotation. They had the Three Sisters and on and on and on. Plenty of native tribes were able to sustainably garden for thousands of years, it looks like, you know, when you look at archaeology. Right. And they didn't overuse their environment. They had as many kids as they wanted, on and on and on, you know.
You know, seasonal food, they would move around, just change, you know, mix up their seasonal food groups and stuff. They had a really good functional plan that, you know, so for lack of a better term, we could steal from Cheech and Chong, and we harshed their vibe by showing up.
Right.
Hey, I'm friends with Tommy Chong.
I've interviewed him a few times too. He's hilarious. That's who he is. But
I'm just giving shit.
But we could relearn, as Steve mentioned earlier, relearn a lot. The past 2 centuries, we've done so many advanced societies, so many ways, but there's only the past 20 years, we started really thinking about what have we done bad that we have to undo. And, you know, the chemical farming, the depletion of soils, the destruction of habitats. I mean, we have to rejuvenate it or it's gone.
And it's not as complicated as people think. I mean, I grew up in North Philly, but my grandparents had probably as far as I'm aware, the only 2-acre size parcel in the whole neighborhood, right? So the whole back half of their yard was 1 acre of garden. And they grew up during the Depression. Right. So they only knew how to do basically what we would now call like natural farming. We would take tomato leaves from the early on, as the plants grow, you take the lowest leaves off just to help the tomatoes accelerate. Same way you do cannabis, right? You kind of top them a little bit. We would take those, put them into coffee jars and ferment them to make sprays that we would then spray on all of the plants and the grapes to keep away the Japanese beetles and some of the other things that would attack the garden, right? We would pick willow cuttings from fresh willow growth and steep that in sunflower oil to make cloning gel so that we could clone the peppers and the tomatoes. And like, this is all stuff that, like, I guess nowadays would be considered natural farming. But if you just go find some books from the second half of the 1800s and early 1900s, kind of pre-World War II, you're going to learn a ton about the type of stuff that's going to be heavily relevant to the current regenerative movement, especially if you can find some old scans, maybe online and things like that. You're going to find a ton of wealth that's not been mined yet in this area as well, that, you know, things that we still don't understand yet. And I think we need to really go back and look at the literature from the 17 and 1800s regarding agriculture and look at it with the fresh lens that we now have and the openness that we now have, trying to get away from a lot of this chemical stuff that we've seen do such devastation, be it aquaponics or living soil or anything else, and what the applications of some of those different inputs could be that they already had down to a fine science. And have been and stuff that people spent 1,000 years working on. I
mean, this isn't a joke, you know, like, how do I say this? Again, the conversation always somehow revolves around Clackamas Coot. He was into artisanal bread, right? But there's some very interesting properties of artisanal bread. There's a lot of the lactobacillus that we're talking about. A lot of very interesting microbes are indigenous to the grain, indigenous to the organism. The yeast and stuff that you use are very beneficial. Where was I freaking going with that? Dang it. My phone is beeping and I lost my train of thought. Dang it. There was a freaking great point there. That time of the day. Yeah, it's the time of the day where a point just gets like freaking vanished out of my head.
Well, you kind of got me thinking, like, we've got experts on fungi, bacteria, pathogens, virus, like everything you can think of. We've never had someone on about specifically yeasts that were plant beneficial.
That would be a good one. Yeah, that would be a good one.
Anyone even know of a specialist that is down? Like, that's such a weird,
for making beer and bread,
right? Like,
because that's another one. Now that we think about it, most people sterilize any kind of yeasts anywhere. You know what I mean? Like, it's gross, but to think about, but with humans, but like with soil, if you ever have any kind of, like, you know, an algal bloom or like a yeasty bloom or something, if it smells weird, you would totally sterilize.
Right, but a little bit of yeast is highly beneficial to a quality lactobacillus collection. If you do that rice collection, it's one of the things where I still do recommend adding a little bit of a rice collection to your traditional kefir-based labs occasionally to re-inoculate, is those yeast compounds that are synergistic and will thrive with the lactobacillus. Also have plant beneficial things. Their exudates are also helpful at fighting pathogens, right? So It's not, something that's going to harm your plants at all. It's actually highly beneficial. And I've never heard of anyone that specifically specializes in plant beneficial yeast. That would be a really cool guest to find.
Should they exist?
Yeah, even if that,
I'm sure there, these days there has to be somebody. I mean, how do I say this? It's cocky of us to say like, well, I don't know anyone in America. Maybe there's someone in China that has it, right? Or someone in India or something. We could take a look. I don't know. And these days with Zoom, you can talk to almost anybody, right? How fun would that be? Like literally, how fun it would be to talk to somebody from the University of Jalalabad or something who's talking about soil yeasts or something. Yeah. The show would have to be like 3:00 in the morning, but still. Well, why don't you tell everybody how to find you here? We'll wrap up the show. It's been quite the episode. It's been heck of I wish I could think of what I was trying to get to. I had a great point, but that's basically my show. We'll keep it's not worth it. I was just going to say that's basically my show. Sometimes we go long enough that it'll come to me, but basically we like to have talks pretty much like this. Ladies and gentlemen, if you guys would like to basically enjoy, honestly, this is basically very much in our kind of spirit talk. We'd normally have a couple more people here, but we'd basically be bullshitting about this kind of stuff. And come take a look, ladies and gentlemen. My channel is Fumidor and the Flavors. Same, YouTube basically. We like to do try to have guests basically on Tuesdays. So I try to do a little bit more straight laced on Tuesdays. This last Tuesday we had Dr. Faust. Potent's going to have Dr. Faust on, I think, in another week or so. December wonderful 2nd. Guest. Some of these folks basically are one of a kind. We just basically spoke to the one of a kind Mycorrhizal expert. Dr. Faust is a one of a kind fulvic and humic acid expert. Clackamas Coote is a one of a kind soil building expert. And anyway, lots of folks like that on Tuesdays. We have great discussions with them. We try to keep it low key with them, but we still have some really, really great talks, breeders and so on and so forth. Wednesdays, we're doing kind of game nights, honestly. And yesterday it was whiskey and game night. Had a lot of fun. Saturdays, like I say, brews and buds. Ladies and gentlemen, come take a look. Also, if you guys have any need for seeds, genetic preservation kits, or photos, take a look at my website, fumidoro.com. What's that, Potent? I still think of all the websites of breeders, Fumidor has the best fucking pictures on his website. Thank you. Your website, I appreciate that. Right on, I'll do so. Gotta open up a freaking tab here. Ladies and gentlemen, I built the website. I took the photos. I built the, grew the seeds, did everything pretty much. Come on. If there's any typos, those are mine too. Especially with the anthocyanin creeping up the trichomes into the trichome heads. I think that's honestly some of the coolest pictures I've seen yet of trichome heads. Well, I appreciate it. Thank you, buddy. Here we go. Fumidoroseedco.com. That's the name. You can go ahead and try to wear it out. He might be talking about Morgana right here. This is sort of everybody's favorite. This is the one I sent out first to people for testers and stuff. This is basically the same phenotype that I have behind me, a little bit bigger picture. You might have been talking about the Ruby Jack. You know what? Let me scroll back here. You had an incredible purple inside the trichome stalks and heads. I think it might be this one right here. Here somewhere. This is one of them right here, Ruby Jack. We'll get to it. There we go. Yeah, well, it's the mom of one of my strains.
The amber, yeah, it's the amber, yeah.
God, that's sexy. Isn't that cool?
That must be a really tasty strain.
Oh yeah, it tastes like pink lemonade. This mom tastes like pink lemonade. Great high. That's her actually.
Pretty. Well, I'm gonna have to get a sample down here.
There we go.
Yeah. Well,
Steve and I have to verify what you say is true.
Right? You have to have weed verification patrol. That's what we're gonna have to do.
Right.
That's it.
And it'll take it. It won't take a gram. It'll take at least a quarter pound for us to figure it out.
A quarter pound, high standards here. Holy cow! Extortion.
At least 100 blood.
Rigged. Ladies and gentlemen, I hope you guys take a look. If you guys do. Coupon code BRAINS for what was the coupon code that I came up with yesterday? Oh, the cookie. It was a whole funny story yesterday. The cookie. Use coupon code the cookie for 15% off. Take a look at the package specials. The West Indian Swindle is very popular. 3 packs, special price, and the coupon codes actually apply. So go for it. Throw it down. Be fun.
Good stuff, man.
Thank you, man. Appreciate it. Thanks for having me on. It's been a fun show.
We also, I wanted to quick mention Mr. Brennan. He's the people over at Spectrum King, if you're looking for great LED lights that have, surpassed all the side by sides that we did. We've also run other ones with some other people on the show. Check out Fish Ganja Guy did one against Black Dog and some other stuff that was on the show and talked all about his results as well. So definitely go check them out. We actually have running them here at the house where we're growing here with our license. So
Cool.
great fucking lights. And then he's also got Grow with More. I'll let you talk about it.
Well, the BioCat solutions, we make more. More is a living blend of hundreds of microbial species, predominantly Protobacteria, archaea, and bacteria with some fungal additives to it in a mild nutrient solution. From seed to harvest, it will improve your plants. We have plenty of customers giving us testimonials of them getting anywhere from 5 to 15% more bigger yields, and anywhere from 2 to 5% to 10% on one strain more THC.
So first time I think I've heard somebody talk about commercially the archaea, because until recently people thought that they were just some soil, whatever the fuck, like just like they were sterilizing for fungus. They just thought they did nothing. And recently I've heard that archaea are the most responsible, or they're responsible for the most of the nitrogen cycling in the soil, much more than bacteria. Is that, I mean, I don't know, actually, I don't know that that's 100% for sure true, but how do I say this completely recolors the picture of what archaea and any number of other microbes are.
So archaea are much more adapted to more extremophile type environments, but also they're finding are a lot of things that they traditionally classified as bacteria are actually archaea. The more as they understand it, but remember that just think about this, and we were just talking about how rapidly our understanding of the world around us evolves. I mean, if you go back, Fumi, when you and I were in high school, archaea weren't a thing. They weren't even discovered till the 2000 something, right? Or late 90s or something like that, where they were even classified. Right. So that's crazy, right? So the fact that we're now getting into, getting these to where we could actually have an industrial use for them is super amazing, considering how rapidly these have been even available in the market.
Right. I mean, we weren't fucking around when we said that, like, you can still talk to some of the people that discovered mycorrhiza. I think the concept of mycorrhiza was known about. I mean, how do I say this? I think maybe like some German scientists in the 1800s discovered it with microscopes, but they had no idea what it was. They just knew that it existed. But until recently, the last 30 years is when people understood what it does. Go ahead.
Well, it's like there's a doctor beginning with a W, and I can't remember his name because I'm a bit inebriated at the moment from various substances. But there's a gentleman named Dr. W. I don't remember what the rest of his name was. Back in the 20s and 30s, had isolated THC and CBD and CBN, right? Way before Meshulam and some of these other guys did, right? So a lot of this stuff was done a long time ago, but our understanding of how to make use of that knowledge was not quite where it needed to be for us to do anything with yet,
right,
or to quantify in a way where we got an understanding of it to why it mattered. And I think a
lot of times that's what happens. People will discover some subatomic particle or something, but I mean, what are they going to do with it? They're not going to also develop a space rocket or something, you know what I mean? Like, that was enough that they developed that one thing, and then 30 or 40 or 50 years later, somebody else comes along. Oh, we can use that.
This, whether it's science or just depth of accepting, things have changed so much, and really with the technology, we can share this information a lot more easily, readily available. I mean, not to date myself, but when in high school 40 years ago for me, you had to go to the library and open a damn book. You couldn't bring up all this stuff. You had to go library, access, our microfilm, and look through all those things into the machine. You couldn't just type in Google and find something in a few milliseconds. And then back then, if I wanted to learn something and it was disproven shortly thereafter, had to go back and figure out what the new thing was this year.
Remember, I was kind of babbling about this earlier about how, now we have drones in our freaking, practically in your pocket. I mean, literally, you can get some drones that are small enough to fit in your pocket. 100 years ago, we didn't know how to fly. 20 years ago, there were search engines on the internet, but they were bone ass dumb. And some of the best ones were run by like Netscape and stuff that eventually became Mozilla. And Bill Gates, who now everyone loves, he did his world's best impression of the biggest shithead in the universe. He bought up or crushed or just destroyed every possible corporation, including Netscape and everything else. And so any of the good search engines would get like hammered. And then Google came along, like basically almost nothing searched for relevance. Then Google came along and ranked everything basically for relevance with their magical little algorithm, right? And so they would actually give you like quality results. So when you'd search for example, Mycorrhiza, it wouldn't give you like 8 billion results that are completely just all over the place and you have to sift through them. It would give you the stuff that was the most relevant in the first place. And then second place, third place, fourth place. It was a fucking revelation. And, and we're used to that now, right? Because like Google's been around for a while now. But that was a crazy thing.
That's just the past 20 years you're talking about.
Yeah.
Remember Ask Jeeves and Yahoo? And it used to change every 6 months.
Yeah, Yahoo was one of the big. Yahoo was the biggest website in the world for the longest time.
AOL search and fucking an AIM. Remember AIM?
Instant messenger, sure.
Ask Jeeves, yeah, straight up. I remember Ask Jeeves. My goodness.
All those things.
And Winamp. Remember all the Winamp?
Right.
I think kind of before YouTube, that was really the way to watch live video.
Right.
That was this is the first way I can remember being able to live stream anything. And I remember watching, people streaming, The Simpsons and like, old cartoons and stuff and weird Japanese TV shows and shit like that on there, and different like religious groups trying to push their religions on there, and all kinds of incredible internet radio, and like, man, things have changed so much.
It's crazy. Well,
especially someone that basically spent the second half of his high school years in a cyber cafe.
That's pretty funny.
Oh yeah, half of my life was. Anyways, at some point we should talk about how I used to basically live the life of grandma's boy, except it was a cyber cafe instead of a software development company. But most of the other things happened.
That's pretty funny. That was a pretty funny movie. The guys on my show recommended. I watched it. I had never watched it before. All right, fine. I think I even rented it or bought it or something. I think I bought it. So anyway, watched it. It's very funny. Very funny.
Very much a period piece.
It
but
is.
If you live through that, you get it.
Yeah.
But yeah, a lot of people don't know when anyways, when I was a teenager, I spent 15 through 17 or 18 and working at a cyber cafe and among other things. It was pretty fucking awesome. A lot of good memories for sure. And back when you could run an internet service provider in the back of a cyber cafe on a random main street. Can't do that shit now.
I just think of the bandwidth.
Yeah, you used to do IT too. Yeah,
No.
I forgot about that. He used to do the same shit.
And I was actually an IT director of a mortgage banking company in the early 2000s until that went to hell. But technologically, what we can see under a microscope, what we can hear through audio, the availability is exponentially greater of what we have access to. So as all this kind of mental acceleration is happening as we're being flooded with data, I'm very curious to see what the mindset is of somebody born today in 20, 30 years, what they think is, that our stuff will be completely outdated to them. It'll be a whole new turn, which is speeding up, I mean, the first computer took it, years to do anything valid with it. And then 30 years later, there were computers that were faster than humans. And now it just keeps going.
AI is coming. Yeah, straight up AI, quantum computing, all that stuff is going to be a huge revolution. And then now it looks like the space race is heating up again, right? The private space race. So
the sad private space race is heating up, but so is the military strategy space race for subsonic new weapons.
They trap us all in a fucking dying Earth by basically destroying all the satellites. Yeah, that's right too. Kessler syndrome. That's a good one.
He's playing a Winamp commercial.
But
remember
Shockwave? Remember?
Shockwave
Macromedia.
Macromedia. Yes,
and installing new versions of Flash and hoping it wasn't some version of like
And Java.
some
and
kind
everything.
of phishing software in it.
Right. That's it.
Kazaa and LimeWire and all that fucking peer-to-peer shit back in the day. And
Oh,
what was it? Net plus plus or something plus plus. What was the old one?
Wait for when the IRC chat. No,
Something
we're talking
something
about
plus
file
plus.
sharing.
I think it was RRC. Fuck. That'd be interesting. How many people currently watching the podcast could even like connect to IRC? Better yet, like do anything
Yeah,
functional.
that was where the dark business happened.
Yeah, that was that. Yeah, for
That's
real.
what-
That was some
that's what-
shit went down on IRC
Yeah.
back in the day.
And
That's
that's,
funny.
you could think this ridiculous minuscule little chat thing, but a lot
It wasn't just Counter-Strike matches. There was
Yeah.
much better shit going on too.
some
If you knew where
pretty
to go.
intense negotiations for other things happened there. Yeah.
And you knew a guy about a thing on the IRC.
I'm trying to remember how much time I spent chatting with people like in just random text rooms on the internet, like 25, 30 years ago on like Prodigy and whatever. I don't know. I'm thinking random stuff, but
I was working at over a cyber cafe and computer repair shop when the 1 gigahertz AMD chips came out. And I don't know if you remember, but there was like a bunch of shitty companies that would put like heat sinks that were nowhere near qualified to take the
They
heat
couldn't
off of
handle
those things.
load
Because
of them, yeah.
those 1
They
gigahertz AMD chips, if you didn't have a good aftermarket cooler on them, man, those bitches would melt
They
the
crack.
motherboards. And I can remember at least
die would
a
crack.
dozen or two things where the chip was perfectly in good condition. Everything else was just melted to fuck.
It would split. It would the damn chip would actually crack. I mean, on that, what the hell was that first? It was the AMD 2s, I think, at that point.
Oh yeah.
But I mean, we used to buy these. I used to build custom computers and for gaming machines for people and put all the extra fans and super cooler things in there and modify them. Even had water cooled ones at one point. That was just what we did.
Well,
because
the
processor, if you overclocked it, would blow up if you didn't have that.
And to circle this back to weed, my very first personal weed grow was a very tall, 3.5 to 4 foot tall gateway computer case that I had modified into a carbon filtered
cabinet.
grow cabinet
that's pretty
that
good.
my mom had no fucking idea that I was growing it. And if you saw my bedroom when I was growing up as a kid, I had as many salvaged and secondhand computer parts as I could get my hands on, and I also do computer repair in the neighborhood. So if someone had a totally fucked computer, I would just rip it apart and use it to fix other people's computers or fix my own or repair my own or whatever. So I ended up hoarding all these parts, and so I was able to kind of become the neighborhood repair guy for a while. And I just always had tons of different things. And then I had fish tanks and terrariums and reptile tanks around, and I would breed the snakes and the reptiles to bankroll all my computer shit, because breeding lots of different snakes and lizards and scorpions pays pretty well, right? You especially if you go to the shows and things, you can make a pretty decent buy. I mean, I made thousands of dollars a month, at a pretty good age where I was able to financially support my computer habit. So
I used to build the game machines for people. I wasn't even a gamer, but I understood how to
do
do it.
that too.
And network the server and all that other stuff. And like spare brother Joe, we're gonna have a gaming party. Like,
If you
what
guys
does
want
that
to,
mean?
if you guys want to look this up, I used to work at a place called Cyberspace Matrix in Hatboro, Pennsylvania. It was a real cyber cafe. There's not that many cyber cafes left in the United States, but there's a couple left in Oklahoma. I've seen one down in Durant. There's one up in Oklahoma City. There's a couple around, but there ain't many. But they're fun, man. I love that atmosphere and that just nerdiness. Maybe
That's like Happy Days on TV. It's just a different lifetime ago,
right?
It's like
It's so weird.
the world's changed that much, and it's speeding. As I mentioned, it's speeding up.
But it's funny. I think one of the funniest times I ever had in a cyber cafe actually was in Iquitos, Peru. I was at the time working in the cyber cafe, and then I went down for a trip to Iquitos to go do reptile collecting and fish collecting and stuff. And I come back, or I was in Iquitos, and on the weekend between the two trips, the reptile trip and the fish trip, and there was a cyber cafe, and I realized all the kids are playing Counter-Strike. So I ended up basically working that as a racket. I didn't pay a dime for any of my drinks or anything else there because I just fucking worked them. Yeah, dude, LAN party. There was a GXL. That was the other one. The first LAN party in the United States that had more than 300 people I was at, which is the GXL, which is a big LAN party. I think they still do events to this day, but 100% supports local fire departments. So they
That's
basically
funny.
take a couple of the fire halls and they rent them out, install them. They have the Wi-Fi that supports high traffic and everything, and the power and all the rest of it that you can actually host those types of events at. They run one of their trailer generators to power the whole thing. And then they host them as a way to raise money for the fire department, which is fucking awesome. I can always get behind the guys that are trying to help us, you know, be it growers.
Especially the fire department. I think fire department has a pretty good reputation most everywhere. You know, they don't fuck with
When
people,
you
and they
ever
just
like,
they kind of
no, the
put
fire
out
department
fires.
showed up. Usually it's, oh
Yeah.
fuck, the cops showed up.
People are rooting for the fire
Okay.
department. Please show up. Please show up. What was I going to say?
Speaking of which,
I was going to say
I
something
don't wanna get
else.
too
You
much
know, like
into it, but if you haven't seen the video in Pennsylvania, like, holy crap! If you thought that some of the more recent events were bad, that was real bad,
off the
charts bad. I'm not gonna get into it because I don't want to get into it too much, but there are few things have sent me to instant rage as much as that video I saw today.
Zero justification.
Yeah.
No justification for that. So hopefully we can finally get some proper law enforcement reform. I think that's something that is needed badly in this country, especially if we're looking towards probably a Republican the next time we have another election, because the Democrats don't seem to do shit when they're actually elected to
they did
office.
just pass that Build Back Better, but they're so bad at messaging that they're basically not even telling anybody.
And again, this goes back to they did a bunch of stuff around health reform that has been game changing from thousands of. I mean, anyone that's under the age of 26 that's still under their parents' health insurance has Democrats to thank for that. You know, but they're horrible at messaging, and we need a third party that's sane and actually gets shit done.
That's the problem. They don't want shit.
The two-party system has to die for America to survive. And it's just that simple.
They don't want.
I'm not getting into left or right. At this point, pox on both houses. We need a new
People
party.
from outside the world or outside the country look in, and they're just kind of horrified by what they see, or by what they see, and they see a very unfunctional government. That's something that people repetitively say. Like when you're inside a problem, you don't always see this. Let's say if you're in a bad relationship or something else, you might not see that, right? But people from the outside, they're like, why are they always screaming? And so basically they say this, on Reddit and everywhere else. If you have foreign friends or something, because they get our news, right? They get our TV, they get our movies, they get our news, they read our newspapers, on and on and on, and they're just fucking horrified by what they see. And like our government is this like non-functional, do nothing, like permanent war thing. It's a very bad combination.
Yeah, and not only that, but just like I feel like we're watching like the death of Rome, right? Like the capitalism has reached its final conclusion. The balance has been shifted past the point where the haves have too much and the have-nots don't have enough. And the equation is broken now. And it's and if you strictly look from a mathematical standpoint, every other time there's been this amount of a mass of wealth at the top compared to the rest of the population,
Inequality, yeah, that's it.
There's been inequality to the level that we have now. There's been revolution throughout history without 100% of the time. And I think that basically the United States is going to go through that. I have no idea what that's going to look like. But I 100% think they're not going to be the same as they are now 10 years from now. And what that's going to happen as, I don't know. Whether or not I even stay in the United States through all that shit, I don't know. If it gets too bad, fuck it. I'm not really tied to the United States enough to where I give a shit. I'm just going to leave. I'm not going to get involved in whatever stupid bullshit. I'm just leaving. Fuck it. I don't care. I used to be all hot and bothered. Why? Why should I risk my own personal well-being over that shit? I don't give a fuck about a piece of dirt. Why should you?
Not only that, but the idea that you would be forced to kill your fellow people, you know what I mean? Or
I know, but no, especially after traveling the world as much as I have, it's not worth it. Life is so precious, and we there's no reason to fucking fuck with people on that kind of level over that kind of thing. It's so trivial. It's so stupid. I just, why? I can't fathom that. So
the only logical answer is greed. It's not a good answer, but it's a logical answer anyway.
I'd rather just not participate. Fuck it. Let them. Anyways, run to the hills is the story.
That's why I've been saying this on my show, that there's this sort of zeitgeist awareness now that some shit's going to go down. So a lot of folks are basically trying to learn how to grow their own food, and a lot of folks in the chat probably are either sitting on their own nice piece of land or looking for one piece of land. So, it might be paranoia, that certainly happens throughout society, that people just basically through paranoia set themselves back. But
the Roman Empire fell
Every empire falls.
due to nepotism, but they fell because people living in the city were becoming sterile and insane, and no one could understand why. They thought that was just the gods going against them, or God going against them as they become Christian at that point. And what it was is they made lead pipe plumbing.
Yeah,
And they were poisoning themselves, so the people didn't get healthy till they got the hell out of the
They
cities.
did even worse than that. They sweetened their wine with lead. They even used like a phosphate lead to do it.
Yeah, no, you could definitely point to
So,
the,
you know, the tipping
cultural
point,
mass
right.
hysteria to lead in the Roman Empire. But in the United States, we have the social drug. We have fucking social media. That's the modern lead.
No, Facebook is unironically responsible for a number of genocides. Like there have been genocides arranged on
Southeast
Facebook
Asia,
groups in
fuck
Burma.
yes, there have. Yeah,
People might think that
Southeast
I'm
Asia,
joking, but if you look
millions
this
of
up,
people
this
have
is a
died because
documented
of
thing.
Mark Zuckerberg.
It's really gross. He had they probably still have it, but it was well documented. He had a chant, company over country. And I can't comprehend how somebody could say that who isn't a psychopath.
The best example is Myanmar. If you really want an example of where Facebook is the dead fucking example of that shit, Myanmar, Facebook literally led the fucking genocide, was on Facebook as for their main communication channel.
Well,
and the worst thing is that they didn't even shut it down. So Facebook, they basically have no customer service. They're $1 trillion company that has no customer service willingly. And they were alerted that something was going on and they just ignored it. Because their little moderation team is over.
It affected their profit margins. They don't care what the content was. Truth is no longer, what's the word I'm looking for? It's variable by perception, alternate facts. You know, the actual truth is now so manipulated, and people
to them, truth is irrelevant. To them, the only thing that's relevant is how many ad click-throughs you basically lead to. You know what I mean? That's it. Right. And so whatever generates more ad views or ad click-throughs or anything else, that's going to be the thing that they push to you.
Whatever maintains their status quo is the path they're going to push as the truth. You know, our political mess we have is the same thing. The truth is no longer valid. They just go spew what they want, keep saying it, and a certain amount of people will believe it.
T. Barrington is asking a relevant question: who's worse, Facebook or Travis Scott? And I was just like, I don't know. It's a little bit like, was that the dick butt, like the reflexive dick butt that has its own dick butt? I'm just like, I don't know, or just like pick something terrible that also has a terrible thing on it, and like that's basically.
Sorry, whose was Facebook or Travis Scott or Scatola Granola? I'm sorry, did I mention?
That's a good one. That's a good one. I think Travis Scott's worse than Scatola though.
Yeah,
he's responsible for multiple deaths, I think.
All right,
then
he
again,
wins
you never
the
know.
Scott battle. By the way, I just want to say that over half the things that Efron talked about today directly contradict his bullshit.
We didn't even ask him about microbial collapse or prolapse. We should have.
Oh, we should have. I didn't even think of that. When he comes on your
show. Is it
show,
possible that microbes prolapse? We should have asked him. I should have asked him with a straight face. He wouldn't know what I was talking about, though. He'd think I was crazy.
It amazes me that people still, especially with the amount of data that's come out now, especially around genetics, it's like you can trust soil genetic sampling. That actually tells you what's in your soil. But
I mean, Potent, there are televangelists that get like people's entire income, like.
Right, but like they're not ethical though. Like at least like I would like to think that at least
you didn't start your sentence with like ethical. You just said like, people.
No,
like,
but I'd like to think
hucksters,
that he believes that he's doing the right thing. Having met the man, I truly do think he believes he's doing the right thing. That's the difference between him and a televangelist. I'll give him that. I don't think he's intentionally misleading anyone. I think he's unintentionally misleading people.
Well, at least that, I guess.
There's a difference. Intent is what, 3/10 of the law? Or what's the
possession?
5/6 or whatever it is. Fuck it is.
Well,
obsession is a perfume.
3/5 of the law. Whatever.
That's what it is.
Sorry.
Social control mechanisms are always put into place when those who have power feel it threatened. To create an enemy, to create a common cause for people to rile up against. is the typical drama that humans do to maintain power.
I could go on a whole rant right now, but I'll save both you and Fumi on the details.
And you mentioned earlier psychopath. I said, no, he's actually a sociopath, where he doesn't care what his actions hurt or do as long as his money is coming in his pocket.
Mm.
And you'll find that most billionaires, most huge CEOs are literally
Sociopath.
clinically verified sociopaths. They're out
Bezos
to
was
get-
just talking openly the other day, and this is not going to happen in the realistic near-term future, so we don't have to be actually scared of it. But I'm scared about the fact that this shithead is talking about it. Bezos was saying that the Earth might definitely have to. He was basically might definitely have to be depopulated to make the Earth basically like a what did he even call it? Like a range park or whatever. I can't think of the word I'm looking for. Like a national park or something for a selected few. And then basically everybody else will live in space in space factories. So pretty much the expanse, but in real life. I mean, but the thing is, it's not some crazy asshole like a bum on the side of the street corner. You know what I mean? This is the world's richest man talking about it. So,
Well,
I don't-
one of them.
Hold on. I don't think it's that. I think if we just change the way that we live our lives and get away, like, let's just look at the change in how a typical nuclear family unit operates, okay? During World War II, families started, what was the name of it? Victory gardens,
Mhm.
okay? And they were growing their own organic food to feed their damn selves. When's the last time you heard anybody do that recently? Before COVID?
It was a dying animal. It was,
In that kind of context.
yeah.
Didn't exist, right? So you had a different perspective about self-reliance when it comes to self-production of food. And I think that we need to give that kind of power back to the everyday citizen. And until we can teach the everyday citizen that they have that power, the same way that you and I and everyone else watching this podcast right now feels empowered to grow their own fucking plants and provide for themselves, be it cannabis as medicine or lettuce as food or tomatoes as food or whatever the fuck else. Until society understands that they themselves have that same power in their own domicile, we simply cannot get past that, right? So we really do need to be better on the education, especially with childhood education, on the importance of gardening. I think that's one of the reasons why you've seen me talk with and work with child educators and bring people like the Aquaponic Association on that work so heavily with STEM education in schools, and why I donate my time and things like that to the Aquaponics Association and pay to be a member as far as the Aquaponics Association is to help further that type of childhood education that really can inspire the next Jeff Lowenfeld and the next Dr. Efron and the next Dr. Faust when they're fucking 7 years old, right? Fuck when they're way older. And I cannot be grateful enough to have all these amazing, intelligent people on my show, but let's breed some new ones and inspire some new ones that can help get us to where we need to be in the next generation and build upon the wealth of knowledge that these guys are so grateful and just generous with sharing their knowledge that we've built upon for decades. Where are the new ones? And it's one of the things I really am so grateful about seeing in Generation Z and the next kids that are even younger than I am, is that they're super passionate about self-sustainability and microbes and all this other stuff that I don't really think of people that are a little bit older than them actually were either. You know, there really are more.
Ironically, that generation is locked out from housing, aren't they? You know, like housing prices now are so excessive that basically most of that generation rents. So now this is a perpetual problem that I hear about, basically, that I hear from. Like, I have a sister that's a bit older or younger than me, and she can't afford to buy her own place, you know, but she's interested in gardening and, you know, vegetables and, you know, fresh produce and on and on and on, you know, and this is a perpetual problem. Like, how the fuck do you buy a house? It's $500,000.
Earlier, your economic reality dictates what you're willing to do. You know, we got lazy, you know, and I'm a bit older than you guys, and I can say that my generation that very few people were doing anything with their hands. you know.
It's even worse than that, Brandon. I was going to say, you reminded me of something from earlier. I had a friend. He's probably, I actually have kind of lost touch with the guy, and so this is a lesson. I should probably get back in touch with him for been a few years now. But he's, I want to say, probably 65, maybe even 70 by now. To be honest, he grew up basically he told me like they lived up in the West Hills. You know, back then it was a little hoity-toity. Now it's super hoity-toity here in Portland. Anyway, but back then there used to be a bunch of German, Italian, and French bakeries run by actual German, like he told me, I don't even remember the names, but you know, Fritz so-and-so and blah, 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 Pierre, blah, 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 whatever. Those bakeries one by one went out of business because the parents, even like these, you know, wealthier parents that lived up on this hill, would go and buy Wonder Bread, literally Wonder Bread that's like liquid dough that's squeezed into a mold and then baked, because, oh my God, the future. And literally that's how they sold it. They had commercials and the little sewing groups and whatever. This is the future of bread. And they basically, like, these old breads that were literally better in every way, better for you, like literally healthy for you, that it cause you to have diabetes, not filled with, you know, complex carbohydrates. Oh, no. Basically everything filled with nutrients and vitamins and ancient grains. Just everything about it was better. They replaced it for something that was objectively worse for really no reason other than marketing. You know what I mean? Just marketing wank. That's it.
Mm-hmm. Straight up for marketing purposes.
Fuck. Stupid.
Again, that's another part which changed drastically during our lifetimes. I mean, they went from being 2 or 3 channels on TV to being who can count anymore, and all that new content, new sources of content. And how do you filter what's true, what's accurate, what's valid, what's fabricated? It's mental saturation. And with people who live looking at their phones and social media, they could easily be led to the slaughter.
Oh, yeah.
You know, because they don't know how to actually do anything. And to see that there are people now, teenagers and young 20-somethings, that want to, you know, get back in the earth and do things. It instills hope for a future. But Steve mentioned, and I don't like saying this, but I believe it, that it's going to take something so horribly drastic to shock humanity and limit the population, because this planet can only sustain.
Population is actually good already. It's just the humans are terrible at cooperating with each other. You know, the problems basically are foreseen are problems. Well, we are going to get squeezed basically from the margins. You know, like basically when you have I don't know, imagine a big old box filled with 100 rats. If you squeeze in the edges and make it smaller and smaller, they're going to start fighting. We are going to get squeezed basically because the coastlines and stuff are going to get flooded, and there's going to be tsunamis and everything else with climate change. But realistically, the population is actually foreseen to drop. So like the earlier, you know, in the mid-century, they're like, oh my God, there's going to be 30 billion people on Earth. Actually, at the moment, there's going to be less. Like China is over depopulating. Like they were excessive with their one-child policy. Now they actually want people to have more and more kids because they're foreseen to drop dramatically in population, very much like Japan. So how do I say this? Like the population problems aren't the problem. It's problems of like water, natural resources, food, equitable food. You know what I mean? Like people that are barely surviving and are basically living in poverty. That's not sustainable. On and on and on.
Our way of living is not sustainable. So unless we come up with these new technologies and come up with a different mindset, I mean, the cost of real estate is insane all over the world now. It's not here; it's ridiculous. It's you know, but if you're a 25-year-old guy with a great college degree and everything else, that doesn't mean you can afford a place. I know, like in Silicon Valley, the young bucks, there's like 10 of them in a house, and they're making 150 grand a year. 10 people, and they can't afford their own place.
That's it, because the rent there will be like $8,000 for a house. Yeah, straight up.
Yeah, but let's look at like how things have evolved. Maybe let's just think about 15 or 25 years in the future. They might have mapped out all the microbes globally that are plant beneficial, right? And narrowed it down to, you need this for this nutrient and this for this nutrient and blah 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 blah. And then you can buy that at the store now, and it's put on any fucking crop, and it's going to grow gangbusters, and it's going to yield better than anything that we can even fathom right now, because they've GMO'd that too.
They've already proven that we've lost microbial species in the past 50 years.
No, I know that, but I don't think they're necessarily lost. They're just not
on
the things that
no
we're
longer
trying to find.
no longer available to find in where they check.
And you know what? Some of that is myopia, actually. I wonder, I hope this is a good analogy or whatever, analog or something. I was just reading today on Reddit, basically, there was something that pops up every once in a while about native bees and how they populate dramatically more effectively than honeybees, but all of our attention is always to honeybees because, I mean, honeybees are cute and they create honey and they're very agriculturally useful and on and on and on, but no attention or very little attention is paid to native bees. They're solitary. They don't have hives. They don't produce honey. They're not cute and adorable, but they're far, far, far more effective at pollination. They've depopulated by like 90% in the last 10 or 15 years. And we just kind of ignore that because we focus on these other things. Evidently, even honeybees are not endangered because they are agriculturally viable. So farmers literally take them from place to place to place, but the native ones are endangered. So
I also wanted to talk about something in chat I wanted to mention. If you are watching this show live, currently the longest lunar eclipse in was almost 600 years is tonight at 4 a.m.
If you have clear skies.
Eastern, 4:03 a.m. Eastern. You can figure out the time for yourself.
It's projected to go for a couple hours. Yeah. So if you have clear sky, go out and take a look. I think it's fucking rainy here, so I don't think I'm going to see it, but we'll see.
Yep.
Might, maybe, you never know. I mean, that's the nice thing. It's not for like 3 minutes like it oftentimes is. It's going to be like a partial eclipse, like, but 90% partial. So still it's going to look like a pretty much an eclipse, but for a few hours as the earth just kind of passes over it with a shadow.
Was it 586 years is the longest one or something like that?
How the fuck do the flat earthers explain that? I'm just honestly genuinely curious. So it's a round shadow. I guess it's a flat shadow, I guess they would say. I don't know, maybe.
That's a great
Everybody's
question.
shadow's flat. I'm not flat. My shadow is flat.
Never thought about asking a flat earther about eclipses. I feel like eclipses kind of instantly disproves the whole.
I was talking. A friend of mine and I were joking about some because she knows a flat earther, and we just burst out laughing because I just didn't even know.
I mean, one of the best underground growers I ever met is a flat earther, and I can't quantify how someone that has such an intricate high level understanding of soils
I know
could not understand physics.
I mean, I guess in theory, the moon could spin around a flat Earth, but why? Like, why? How and why? Also, would the moon be flat or not? I don't know. Maybe.
Does it spin around both sides of the flat Earth?
You know what? It's convenient because we always see the same side of the moon because the moon is actually geospatially locked with us, so we always see the same side no matter what. So they would tell us that the moon is probably flat too. I bet.
My other question. Someone bring this up in chat.
Like
stare at plants. Thank you
plates.
for this question. How do flat earthers explain time zones?
Basically, it's like a plate, and part of the plate is where you put the peas, and part of the plate is where you put the potatoes, and part of it is where you put the chicken. That's the different time zones. That's what they would say, probably. I'm making that up, but
a giant like black piece of pie spinning around the earth. Like, I don't know.
Well, if the sun comes up here and goes down there, and it does it,
the tide goes in, the tide goes out. You can't explain.
That's the thing. If it's a flat Earth, why does the sun spin around it? That doesn't make any sense. Why doesn't the sun just go back and forth like a barcode scanner?
Yeah, but how does a flat moon, move in an arc over the flat Earth?
No, but you know what I mean? Like, why? Why would there be a sun? Like, anybody that has a theory, like, I mean, why does that stuff work? How does that stuff work? Like, how does a sun orbit around a flat Earth? Like, so somebody built that, right? Okay, but so then why did they build it that way? If somebody built that, because nature didn't build that, because nothing works that way. You couldn't possibly find something to explain that. So it has to be some kind of a creator. So if that creator built that, why wasn't the creator smart enough not to have the sun just go back and forth instead of like spin around? Because if it's a flat Earth, you just want it to go back and forth and back. That'd be efficient.
Do over.
Why is
it's like a light mover? Basically, you'd have a light mover. You have the sun under a light mover.
If the sun provides life, why do we have night?
Exactly. And not only that, why does the sun have to move back and forth? If you have a flat, just turn it on, turn it off, like a light switch.
Yeah, exactly.
This is a dumb creator that they have, basically, in their flat Earth world. Their God is the dumb God. I'm calling it right now. That's the dumb one. Like everybody else's God is super smart. That one is super dumb. That's the dirty one.
I had someone giving an interesting response to that one time, and he was like, well, what if like he was spawned into existence when the universe was?
That
like-
God is the little Nicky of gods.
So he's
He
kind
was
of
hit
like
in the face with
learning
a
as he
shovel
goes
by his other
along,
brothers.
learning as he goes along. That's why we have an asteroid belt that he fucked up the first time.
That's it. That's it. The asteroid belt is predicted to be not one but two fucked up planets.
On which day of the first 7 or 8 was it that he had a
It was
bad
like day-
day? He's
yeah,
like,
day-
"Fuck that up."
Right?
Yeah, day 6 he fucked
Fucked it
up,
right
so
up.
that's why I take the day
Do
off.
over, crumpled it up, fucked that up too. Like he didn't even crumple it up well, right? You're supposed to crumple it up and throw it in the garbage can. He just left it.
Edited out day 8 where he remade everything.
Just left the trash there. Didn't even bother to clean up his homework. Freaking his mom's gonna be so pissed off when she comes back. Then Jupiter's way too big.
His mom
What the
can't
fuck?
come back.
Jupiter is like bigger than all the other planets combined. You could fit all the other planets, including the second biggest planet, Saturn. All of them fit into Jupiter, and you can rattle it around.
Isn't
The
it
fuck?
1,000 times the mass of Earth or something like that?
I think it's like a million
I
times.
always-
It's stupid.
So I was raised Southern Baptist, and I always that in my head, that's how I always when people explain to me they're like, well, Jesus took off and went into the sky, and I was like, so he's like
Did
tripping
he land
around
in the
the
giant red
universe,
spot?
just like dropping into random planets, is like a hippie preaching fucking good vibes, and then peacing out to the next one, right?
That's it.
So like that's.
Unless
I
you
can
show him.
totally envision like an omnipotent being just like creating this crazy universe with all these random like procedurally generated planets and then bouncing around trying to make everything better, right? Like
So they're all flat though. All those Earths are flat. So how does it get there? Like
If he
Think about-
made that one that way, but the other ones might not be because he may have made them differently.
the
Right.
other ones are triangular or something.
yeah, because
That
each
was what I
one's a
just
little bit
mentioned.
different. The chemistry is different, so he's got like a new thing that he has to like make it different each time. Like he is, it's a
Hmm.
challenge, right? It's kind of like if you were making a universe, you'd want to do that. Like that's how kind of sounds like a fulfilling, fun.
They're kind
thing.
of fun. I just I downloaded Civilization because we've been doing game night on my show. So I played a couple civilizations. I started as India. Gandhi is very warlike. Then I was Norway. That was kind of weird. Everything's beards and Viking ships and stuff. I guess it's like that, you know. You can basically try a couple civilizations and just abandon them and then move on to the next one.
My teenage understanding of Jesus.
That's it. Like this civilization is boring. Like I don't have enough cities. Like Egypt has way more cities. I'm gonna just try another one. I'm gonna just see if I can get France.
Some topics that I
Then he moves
tried.
on to another solar system. This one has 2 suns now. They orbit in fun ways. One is yellow, one is orange. It's awesome. I like this. But I like that better than the flat Earth God. I still think he's just basically a
Right.
fucking idiot. Honestly. Like, why is that sun not on a light mover or just on a light switch? It's so much more
my
efficient.
understanding is- Okay, so let's just put aside the like all the other obviously broken bullshit with the concept. If you have let's just look at it this way because we're going to use some random objects here that I have at the table. So we have like some rosin press.
Everybody on Earth runs to one side. Does it tip over?
Right. So if the Earth is a rosin press bag with hash in it and rosin, okay. So let's just say that this is the
I
Earth.
know what you're going with. Yeah.
So now we have this flat moon. How does the moon know without a spherical gravitational pull to stay in line? Wouldn't
That's
it
the man in the
like-
moon.
Wouldn't it
knows.
want to be like this and then like tilt as it goes through the sky to be like this gravitationally and then tilt like this?
Well, it's the
doesn't-
man in the moon, and he has it's a giant rocket ship basically made of cheese, and he pilots
I'm
it.
trying to prove physics and how broken
That
flat
doesn't
Earth
but-
is
but the
with rosin, wax paper, and a
peanut butter
peanut
cap.
butter cap.
And that probably makes
Why would the
more sense.
tides sometimes be up and sometimes down when you know that the moon is that way and not underneath the Earth?
I mean, this is funny because we're talking to 3 people that have an understanding of physics, but I don't know. It's just funny to me that someone can possibly think that any of that is true.
You know, and they're not pulling your leg at a party like they
No,
need
I
it.
know,
I'm
I've
like,
met-
Where does
these-
Santa Claus live if it's a flat Earth? Under the Earth or what?
the north edge.
Probably. All right. Yeah, like right at the edge, probably right next to the ice wall, I guess.
The
'Cause
top
there's
of the
apparently an ice wall. Because I'm always like, how does the ocean not fall off? And then, oh, there's an ice wall. All right.
It does fall off. Where does it fall to?
I mean, that's what I would think, but I mean, like it's like it's temperate. The middle of the ocean is like warm. Like, how's there an ice wall? Apparently, there just is. It's very convoluted. Like when your theories are that convoluted, you have to wonder if they're plausible. But they say they are. So
I don't think anyone thought any of this
believe
through.
them.
Here's a question, and if someone can answer this that's listening to the podcast, please email me at ponics@gmail.com. Can someone explain to me how gravity works in a flat Earth world in terms of gravitational pull if a moon is involved?
It's because it's just the Earth loves you and doesn't want you to leave.
Tide goes in, tide goes out. You can't explain
Can't
that.
explain it. Can't explain it.
No, they can't explain.
No, I get it. We're both. I think Fumi knows what I'm talking about. We're mocking Bill O'Reilly because he's a fucking moron.
Yeah.
But
Mr. Lufa.
the one thing I ever want to say, if you're ever having stage fright, I used to think about this when I was doing stand-up on stage, or if you're ever doing like a motivational speech, you're talking at work, you're worried about giving a speech at a eulogy or a wedding or some other thing, a bar mitzvah, whatever. Always think about Bill O'Reilly losing his goddamn mind, thinking, oh, you know what does he mean to play us out? To end the show? Fuck it, we'll do it live. And just think about Bill O'Reilly losing his mind over simply plugging the fact that Sting was releasing a new album. And you'll get over whatever it is that's bothering you at the time. Seriously, it's worked for me.
He's just a piece of shit. I prefer, honestly, if I'm in a bad mood, I prefer to watch Tippy Taps on Reddit. You'll find like today there was a little dog, and just the stupidest little 5-second video, but it's just the dog that was super excited. And it's like, just super happy. You just forget whatever dumb garbage you were talking. Like fucking Bill O'Reilly. Guy's a fucking sentient asshole. I mean, come on. It's a walking, talking asshole.
I'm mocking how insecure he was about simply plugging a Sting song.
That's it. He's a sentient asshole.
That's my point. Like the whole concept of the whole event is hysterical.
His sense of importance is maybe disproportionate.
Yeah, you're giving him importance that he doesn't deserve.
The whole thing is fucking fuck you, hysterical. Yeah, that's why it's funny. It's like meta funny.
I know what you mean, but I'm just like, I've gotten over the guy. Like I've heard the name Bill O'Reilly like I don't know how many times in my fucking life.
Yeah,
it's like I prefer to think of like a dog tippy tapping on the floor. So like, we're going to walk.
On the next 3 hours or 3 minutes and 30 seconds because we're about to hit 4 hours, and I do want to cut off the show.
Let's go for
lunchtime. Everybody had to find you, Brennan. More info
here. No. Spectrum King LED, BioCat Solutions, more. I don't see smoke signals so well anymore. The eyes aren't as good as they used to be. So chiseling rock and mailing it doesn't work. Rates are crazy and they don't deliver. But Spectrum King, or you can ask Steve, where's that other ancient hippie guy? And he'll tell me.
Yeah, if you send me an email or something, I'll pass it on to him and get him to reply back to you. So don't worry. If you shoot me an email at either potentponics@gmail.com or Brendan at BRENDAN.
Just at Spectrum King LED or at BioCat US if you want to know about the microbial thing or about LED lighting or
Can people reach you telepathically?
So,
okay.
And it's scary having others inside your head.
Right on.
That aren't your own typical voices, the crowd you feed yourself.
Gotcha.
But very few people read minds contrary to what others are.
But especially if a lighting question and stuff like that, and you're looking to get lights, he can help you get set up with the right lighting for your grow room and all that stuff. Or
whether it's orchids to cannabis to algae on water, the beauty of studying light and its effect on things is something we've done for a long time. So we do take it seriously, and we did start the full spectrum lighting movement. When LEDs first came out, a lot of people had bad experiences because they lacked the intensity. They were blue and red or purple or blurple or whatever else you want to call it. Knowing what plants need and want and getting some of the right intensity levels will get you better results.
How do everybody find you? Fumi?
Pretty easy. Go take a look at my website, fumidoro.com. Very easy. There's even a contact page over there. You can email me at fumidor@chronictable.com. Chronic table, just like it says right there. One big old word with the @ sign, fumidor@chronictable.com. Very easy to find me. If you guys have one of these days, we might get back into trivia. If you guys have trivia questions, or if you guys would like to see trivia again, doing kind of ganja trivia, email me at trivia@chronictable.com. It's also easier to remember conveniently. Follow me at Fumidor_Chibidor on Instagram. And if you guys would like to participate in the Portland Cannabis Tasting Society's Fumi's photo contest, you guys are eligible. Everybody that listens to this is eligible. We just do a people's choice photo contest. People with the most votes win. Throwdown, basically, pretty much every month we do a new one. Throwdown, one post per day over at Portland Cannabis Tasting Society. If you guys would like to know more, just come follow the page and ask me, frankly. It's very easy. Cheers.
Awesome. And you can find out our guest over at MycoRoots.com. Super knowledgeable guy. Email him. He can do mycorrhizal fungi sampling and all kinds of other awesome stuff. They do a wonderful range of different fungal services for testing for pathogens or beneficials, or a whole bunch of other awesome things to better understand your soil. He's a great dude for that. And then also, you can check out myself over at Aquaponic Cannabis Nutrients. If you need newts, we have a bunch of kits you can punch in. Say you have 12 weeks of flower that you wanted to run in a longer system. Pick your aquarium size, pick your weeks of flower, and you can get your pre-weighed packets every 2 weeks, and you can see some side-by-sides with and without some of the different nutrients. So we were going through earlier trials, so definitely check that out. Also, Marty and I have a wonderful aquaponic cannabis class over at apmjclass.com. We have a huge range of different topics. We're constantly adding new content. Our next live session will be on Sunday. Be sure to check that out if you're looking to join us. You can use code APMJ now through the end of Cyber Monday in order to get $50 off the class. So check that out if you're looking to save a couple bucks.
Does the frog come with the class?
Yeah, I mean, if you graduate the class, I will find the frog and send it to you. How about that? But you got to finish it though. Cool.
There he is. Might be a girl. You never know.
I just always like it because we do find tree frogs occasionally in the greenhouses when they sneak through the ventilation and things. And you know what? It kind of proves that we're not
free
using
IPM.
toxic. Well, it's free IPM, but also a proof that we're not spraying harsh toxic chemicals, right?
Yeah,
right.
The fish and the fact that we can even have amphibians in the
exactly.
ecosystem is honest proof that we're not lying about what it is that we're spraying in our facilities.
Basically, because amphibians are like the canary in the coal mine. If there was any toxins, they would just fucking die, right?
Instantly, yeah. They're way more sensitive than fish are. So.
Interesting.
All right.
Cool.
Well, thanks everybody for watching. I really do appreciate it. I'm trying to get an episode up for Monday, a guest lined up for Monday, just because Thursday is the holiday. We will not have an episode on Thursday. We're going to take a day off. I think y'all are going to be busy anyway.
Thanksgiving.
But we will definitely be back the following Thursday with Dr. Robert Faust. He's going to be talking to us about some of his awesome work that he's been working on. He was just on Fumidor's show, and he's going to be talking to us about some of his work as well on our show. So thanks everybody for watching. You can find me on the Potent Ponics on SoundCloud, iTunes, Spotify, Amazon Music. We're on basically everything at this point. So definitely check us out, and we'll catch you guys again next week on Monday. And check out Fumidor's show on Saturday. How do they find you?
Fumidor and the Flavors on YouTube. That's the one.
Awesome. So we'll see you guys over there on Saturday, and take care, everybody.